morning.
So, Dr. Dillan has joined?
Yes, sir. He was there, sir. Just already his video is on, sir.
He is online, sir, but not on the seat.
Not on the seat.
Okay.
Yes, sir, has come.
I just went away to see number of pages of my PhD thesis.
Good morning, Dr. So you're going to ask questions from your PhD thesis, I guess.
I am going to tell a good, an interesting anecdote to the young students that they should not feel.
That's wonderful. That's wonderful.
It must be in the library and they can check.
Yeah, we have maintained, sir. We have.
Yeah. How many big pages are there?
Yes.
Not A4. Full of. Yeah, it all depends upon the matter as is. Makes you work hard and learn something. Yeah.
Should we start, sir?
Yeah, please.
Still we are joined with 123. I guess people shall be joining shortly.
Okay.
The faculty members and the students, many of them would have joined on the YouTube channel. But I wish they should be there in next 5-10 minutes, but we cannot delay the proceeding. Once again, I would like to welcome all of you, the chair of the program, Professor Dillan, and all the jury members. We also have Professor R.M. Singh, former professor and head division of genetics from Banaras Hind University. Out of his sheer interest, yesterday also he was listening, and today also he wanted to listen. So he's also
it's a pleasure.
There. And I welcome him.
नमस्कार सर।
नमस्कार
सर, आपका स्टूडेंट हूँ यहाँ पर भोपाल में। नमस्कार
Dr. Patra, yeah, so the jury has Dr. Patra, of course, he was BHU student in BSc Agriculture. So I think without any further delay and due, I would like to go ahead with the proceeding. Dr. Rasmi.
Yes, sir. Thank you, sir. And very good morning to all of you and warm welcome all of you on the second day of our 60th Convocation Week program. So as you are aware that yesterday we had the presentation by the MSc students for the merit medals and the best student of the award, Professor V.L. Chopra, Best Student of the Year award. And today we are continuing the same process of presentation by the PhD students of their significant research achievements. And I welcome Dr. B.S. Dhillon, Vice Chancellor, former Vice Chancellor, Punjab Agriculture University, Ludhiana, who is chairing and he's chairing the judging committee members, and the jury members, Dr. P.G. Patil, Vice Chancellor, MPKV. I warmly welcome him. I also warmly welcome Dr. K.C. Bansal, former Director, NBPGR, and also I welcome Dr. A.K. Patra, who is a Director, IISS Bhopal, and formally I also very warmly welcome Dr. Rakesh Singh, who is Professor Economics at BHU Varanasi, and I would like to welcome our Honorable Director, Dr. A.K. Singh, all the faculty members and all the distinguished dignitaries who have joined or who will be joining shortly, all the heads and professors, faculty, students, ladies and gentlemen, all are very warmly welcome on the second day, and especially welcome all the students and also the students who are going to participate in today's contest. And with these words, I also would like to say best wishes to all the students. Let them present their best and interact with the jury members, and they should be very confident in giving the answers. In a very short, crisp answers should be given, and we expect more from the PhD students as they have more experience and their research programs are also very extensive. So with these words, I once again warmly welcome each one of you and the jury members and the chairperson of the judging committee and all who have joined. And I think now we have around 205 and now they are joining slowly. So we begin the proceedings so that we are not delayed. So thank you, all of you. Dr. Over to, and I also warmly welcome our convener, Dr. Anil Dahuja will also be the convener today also. So Dr. Anil Dahuja, kindly take the chair, the platform, and again you can highlight the today's procedure.
Okay, thank you very much. So, Honorable Chairperson of the session, Padma Shri Dr. B.S. Dhillon, esteemed members of the judging committee, Dr. P.G. Patil, Dr. K.C. Bansal, Dr. A.K. Patra, Professor Rakesh Singh, respected Director IRI, Dean and Joint Director Education, Joint Director Extension, Directors of various research centers of IRI, Directors of Sister Institutes, Associate Dean Dr. Manjaiya, Registrar, other distinguished guests, HODs, professors, faculty members, and dear students. You will all agree with me that we had an exciting and intellectually stimulating session yesterday, wherein our MSc student presented their significant research findings for 5 IRA merit medals and NAMAD Professor V.L. Chopra Gold Medal and Best Student of the Year Award, which carries a cash prize of ₹25,000 and a certificate. Undoubtedly, the icing on the cake and highlight of the session was engaging interactions of the students with the members of the judging committee, who are the stalwarts in their own fields. I'm sure these interactions must have had a profound impact on the overall persona of the students. Yesterday's experience would certainly change their perception and perspective towards the science and will surely expand their horizon and vision. So without taking much time, I would briefly apprise the audience, especially our esteemed chairperson and members of the judging committee, about the stipulated guidelines for this prestigious award and medals in the PhD category. So there is not much change which is to be followed. Most of the things they are very similar to what we followed yesterday for MSc students. The process for obtaining the nominations from various disciplines remains the same. That is, the minimum OGPA of 8 is a prerequisite for PhD students as well. As far as the assessment of the applications of the nominated candidates is concerned, there is no change as such in the parameter to be used for the purpose of evaluation. But pertinently, the weightage of these parameters is slightly different for PhD students. As I mentioned yesterday, for MSc students, the weightage for OGPA and research paper published out of the thesis was 50% and 10% respectively. But for PhD students, since we expect concrete and tangible outputs from their thesis, the weightage of the research papers published is 40%, while the weightage of the OGPA is only 20%. Here, I would also like to emphasize that only those research papers published from the student's thesis are considered where he or she is the first author, and also the review articles are altogether excluded during scoring of the applications. There's no change in the weightage of other parameters on special achievements for both MSc and PhD student. It is 10%. And similarly, the weightage of today's presentation also remains unchanged. That means the esteemed judging committee once again will be evaluating these presentation for 30 marks based on 3 sub-parameters. The first one is the quality of the research work and its potential impact, which again carries 15 marks out of the total 30 marks allocated for today's presentation. And then ability to answer the questions posed to them, which also has a weightage of 10 marks, while the style of presentation and the quality of the slides carries 5 marks. Sir, the list of the candidates along with the performer for the purpose of their evaluation has already been shared with all the jury members. As per the list, 14 students will be competing for this coveted award and medals today. However, I would like to inform the House that one student from the discipline of bioinformatics has opted out of the contest due to some unforeseen reasons and the circumstances. So the mode of presentation, as yesterday, would again be a pre-recorded presentation, but of course the duration of the presentation would be 12 minutes in place of 10 minutes that we had yesterday for the MSc students. The presentation of the nominated students would be played as per the name of their parent discipline in the alphabetical order. And also the candidates have been instructed to keep their videos on while their presentation is being played here and be ready for the subsequent question and answer session. The members of the judging committee would be initiating the interaction as we did yesterday. However, anybody from the audience is welcome to ask a question or pose a query to the presenter. But I humbly request each one of you to please virtually raise your hand if you intend to do so. I, with the due permission of the chairperson, would be calling the interested questioners from the audience one by one for their interaction with the students. So with this brief introduction, I would now like to wish all the contestants good luck for their presentation and thank each one of you for your virtual presence here. So now I take this opportunity to request the Honorable Chairperson of the session to kindly give his introductory remarks and conduct the session further. So wishing you all a fruitful, productive and rewarding day. Thank you very much.
Thank you, Dr. Dhuja. Thank you very much for very kind words. I assume that all the students program yesterday, so I will not go in detail, etc. First of all, I welcome all jury members, Dr. Patil, Dr. Bansal, Dr. Patra. Dr. Joshi is not with us today. Dr. A.K. Singh and Dean Madam Rashmi Agarwal and Coordinator Dhuja Sahab. Welcome. Good morning, Namaskar. And special welcome and special good morning, Namaskar to Dr. R.M. Singh ji. It's very good that you are participating today and we hope to get some guidance from you. Dear student, I felt yesterday that in some cases we were doing not justification to the student, asking some difficult questions. But today it is different. PhD students. As PhD student, I used to say that I know more than others about my PhD work. And because my guide, outstanding person, everything outstanding, but I was working on my thesis for 4 years or 3 years. So that way, I thought that, first of all, I say good luck to everybody. But if someone doesn't perform well, please don't feel that, oh, something is wrong. This 10 minutes examination presentation, just a very, very minor thing. I'm very sure you must have fully prepared. So because of yesterday's proceedings and because it is very easy to ask questions sitting on this side and difficult for the gentleman who is going to appear for any interview, etc., because he's already under pressure and the examiner just comes with some questions. Many times. So I thought that I will just explain, I will just share an anecdote. Dr. A.K. Singh, because you reminded me yesterday, I was gold medalist. So I thought, let me, you may say that again today, it's good you didn't say. So I thought that let me just to motivate the student, just to keep their spirits high. So my dear student, my external examination was Dr. D.P. Singh from Kanpur, who later on came to HAU and then became Vice Chancellor at Pantnagar also. His, I think, second or third or fourth question was plant type of arhar. And we had studied only plant type of wheat. Dr. Sana paper and one paper before that from you, from I think Netherlands. What was the name of that? I don't remember. Maybe after. Told just normal things of plant type, and he was not at all satisfied. So my viva was not good. Of course, I am talking of comprehensive. Of course, I got through without any problem, maybe because of my academic career in MSc and PhD. I am MSc from PhD, and there also I did well, by grace of God. Now, then came thesis viva. Is 457 pages. I thought that I should check the number of pages. And many pages are double size because of large data. And the gentleman who came to examine me, He was class fellow of my guide. You can see name of the guide from my thesis. I will not name him, a wonderful person. So I was asked to tell important finding. He said by name, called him by name. So much work, is it needed from PhD student? Did you do justice to your student? After that discussion went between them and Dr. H.K. Jain, who was head at that time, he intervened. My external examiner grilled my guide, and I had only explained what are the outcome. So we have some days very good, some days not very good. Don't be discouraged. We are here to motivate you. We are here to support you. But certainly question, introspection, there must. If we say very good, balle, 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 then no. There are many papers how to make good scientists. I will not go in detail. How labs learned, they obtained Nobel laureates. How noble they worked. And one very good example is division of genetics. The chair of the head of the division of genetics, you see starting from B people, I will narrate them. So that way, my best wishes to all. And one few 6% will be winning. Others are equally important. Without participation, there cannot be any contest. Unless until there is one person who is going to lose, others cannot win. So my best wishes to all. We may ask difficult questions because now PhD is the ultimate degree, but please don't be demotivated. Wish you all the best. Best wishes to all. And I think, thank you all.
Thank you, sir. So with your due permission, sir, so can we invite the first candidate for the
Yeah,
presentation?
please. As I requested yesterday, please don't ask for permission. Just move on.
Okay.
Just
sir,
move on with your sweet voice and wonderful English. You speak as if you are singing a rhyme.
thank you, sir.
Yeah, please.
Okay, with this, I invite Ashish Khandewal from Division of Agriculture Chemicals.
Honorable Chairperson, respected jury member, distinguished scientists, and dear students. Good morning, everyone. God has created Earth with suitable environment and plenty of resources for life to survive. But due to increasing population, the demands are continuously rising on crude oil or petroleum-based products. To overcome this demand, crude oil is extracted from beneath the soil and transported to our country. Imported crude oil either transported or refined at refinery to make it usable to cater the need of the population. According to Standing Committee report, more than 200 accidents occurred in oil companies during 2014 to 17. More than 1,000 accidents have been occurred during their transportation. Besides this, accidental discharge of oil due to non-replacement of old oil pipeline occurred on agricultural fields in Assam, Tamil Nadu, Gujarat. In recent year, major accidental cases have been occurred in Assam, and farmland and social life of human beings get disturbed due to contamination of farmland with crude oil. As per US EPA, bioremediation recognized as an alternative technology for permanent remediation of contaminated sites. Therefore, presented investigation on bioremediation of aliphatic and polyaromatic hydrocarbon from oil contaminated soil using microbial consortia was carried out using following objectives: to extract crude oil from contaminated sample and characterization of its various components using chromatographic and spectroscopic techniques, to isolate microbe from contaminated soil and their characterization, to evaluate microbe or microbial consortia for remediation of oil component under laboratory and pot condition and proteomics study, to develop usable formulation of potential microbe or microbial consortia and validation. Under first objective, crude oil is extracted from contaminated soil using soxhlet extraction and shaking and dipping method, and highest yield was observed using soxhlet extraction approach. The isolated oil and contaminated soil were characterized using GC, HPLC, AAS, and elemental analyzer. An analytical method for extraction of aliphatic hydrocarbon were standardized using GC equipped with flame ionization detector. Further, an analytical method for extraction of naphthalene, fluorine, phenanthrene, anthracene, and pyrene were standardized using HPLC. Under second objective, bacteria and fungus were isolated from crude oil contaminated soil. Isolated bacteria and fungus were characterized using 16S rRNA and 18S rRNA approaches, and accession number and phylogenetic relationship for 17 bacteria and 3 fungus were obtained. Under third objective, these minimal medium were spiked with 10 ppm mixture of naphthalene, fluorine, phenanthrene, anthracene, and pyrene in the absence and presence of 280 surfactant, and were subjected to degradation by isolated bacteria and fungus using standardized procedure. Among 17 bacteria, highest degradation of pH was observed due to Cochrea rosea in the presence of 280 surfactant, and naphthalene was found to be easily degraded compound among all 5 pH. Among 3 fungus, all 3 fungus showed similar degradation pattern, and percent degradation of phenanthrene and anthracene was significantly affected by 280. Based on mutual compatibility of Cochrea rosea and Aspergillus pseudobi, medium was spiked with 10 ppm pH mixture, bacteria, fungus, and their consortium. Results showed that microbial consortium exhibited significantly higher degradation for fluorine, phenanthrene, anthracene, and pyrene. During degradation, phthalic acid, anthrone, phenanthrene 910 oxide, 9-phenanthrol were observed, and probable pathway for anthracene, phenanthrene, and fluorine were derived. The work was published in International Journal of Environmental Analytical Chemistry with NAS rating 8.826. Minimal medium were spiked with 1% level of crude oil in the absence and presence of 280 surfactant, and were subjected to degradation till 30 days by isolated fungus using standardized procedure. Results showed that based on degradation of aliphatic hydrocarbons, compounds were clustered into 4 groups. Further, among all 3 fungi, Aspergillus pseudobi showed higher degradation for aliphatic and total petroleum hydrocarbon. The work was published in International Journal of Environmental Analytical Chemistry with NAS rating 8.826. Degradation of crude oil by isolated bacteria in minimal medium were investigated, and results showed that 3 bacteria coded with A3, A9 and AK7 showed comparatively higher degradation for all aliphatic hydrocarbons. Further, based on percent degradation and mutual compatibility between Bacillus amyloliquefaciens and Pseudomonas species, their consortium were immobilized. Under fourth objective, microbial consortium or fungus were transferred to beaker, and bentonite clay and calcium chloride solution were added for development of bentonite alginate composite beads, and that were characterized or observed for pH, size, wettability, FTIR, and shelf life. In second approach, guar gum were added to nanobentonite. Subsequently, 280 were added to microbial consortium or fungus, and it was added to nanobentonite, and were subjected to extruder for water dispersible granule preparation. The doublet formulation were characterized as per CPEC guidelines. In third approach, microbial consortium or fungus were transferred to beaker, and bentonite clay and carboxymethyl cellulose were added. Homogenized solution were lyophilized, and developed formulation were characterized. The crude oil, free culture, and microbial composite were mixed into soil, and experimental soil was incubated in BOD. Samples were extracted over different days using standardized procedure. The degradation data were subjected to first order kinetics, and half-life were calculated. Results suggested that degradation slowed down with increasing oil content. Bio-augmentation of bacterial consortium and fungus reduced the half-life, and bacterial consortium found to be better oil degrader as compared to fungus. Among bacterial and fungal formulation, bacterial formulation performed better, and guar gum nanobentonite composite showed better efficacy as compared to other composites. The control soil and crude oil fortified bacterial consortium or fungus inoculated sandy loam soil were subjected to taxonomic and functional gene copy based study using standardized procedure, and taxonomic and functional gene copies per gram of soil at 10th days were calculated. The result showed the variation in diversity of microbial composition. Addition of bacterial consortium resulted in increased in gene copies of bacteria, alpha proteobacteria and firmicutes. However, fungus inoculation increased the gene copies of beta proteobacteria. Among different functional gene copies, NIF-H were found to be in abundance as compared to other nitrogen-related bacterial gene copies, and nitrogen regulation by MOA and NIF-H bacterial gene copies were found to be effective due to bio-augmentation of bacterial consortium and fungus in oil-fortified soil. The work was published in Frontiers in Environmental Sciences with NAS rating 10.581. Under next experiment, pH mixture, microbial consortium and bioformulation were mixed into sandy loam soil and incubated in BOD. Samples were extracted over different days using standardized procedure. The degradation data were subjected to first order kinetics, and results showed that Microbial consortium inoculated treatment showed better degradation as compared to control. Further, guar-gum nanobentonite water dispersible granule bioformulation showed better degradation as compared to other treatments. The control and treated soil were subjected to taxonomic and functional gene copy based study using standardized procedure. The results showed that variation in diversity of microbial composition and inoculation of microbial consortium in pH fortified sandy loam soil led to an increase in the gene copies of alpha protobacteria and firmicutes. The results of functional gene copies showed that NIF-H gene were in abundance, and increased abundance of MOA, NIRK, NMOX, and microbial consortium treated soil suggested that enhanced degradation might be due to increased nitrogen regulation in the soil. The revised manuscript submitted in Environmental Science and Pollution Research with NAS rating 10.2. Under validation experiment, bioremediation of naturally oil contaminated loamy Assam soil were conducted using selected bacterial consortium of Bacillus amyloliquefaciens and Pseudomonas species and Aspergillus adobe fungus. In degradation experiment, bacterial consortium or fungus and crude oil contaminated loamy soil were transported into pot and extraction were carried out over different days, and aliphatic and total petroleum hydrocarbon were quantified using standardized procedure. Results of degradation study showed that bacterial consortium showed highest degradation, and results were found to be in similar pattern as it was observed in oil fortified sandy loam soil. The DNA of naturally oil contaminated and treated soil were extracted at 10th day, and DNA was subjected to quality check, followed by analysis using ion torrent sequencing on ion torrent platform, and reads were subjected for metagenomics and proteomics analysis. Results showed that across the treatment, microbial diversity were significantly different from each other. Further, overabundance of Bacillus and Pseudomonas in T2 treatment indicating the presence of added bacterial consortium as compared to T1 and T3, and Venn diagram showed the number of unique species over different treatments. Results of cluster orthologous group functional profile showed that bacterial consortium T2 were metabolically distinct from T1 and T3. Similarly, cake orthology functional profile showed the significantly abundance of enzymatic pathways as compared to T1 and T3. In nutshell, method for estimation of aliphatic and polyaromatic hydrocarbons in medium and soil were standardized. Based on percent degradation, Pseudomonas species, Bacillus amyloliquefaciens, Aspergillus adobe were identified as best crude oil degrading microbes. Half-life of total petroleum hydrocarbon were decreased by 5.44 to 6.97 times due to inoculated bacterial consortium. Cocuria rosea and Aspergillus adobe were identified as best degrader of pH. Half-life of pH were reduced to 2.2 to 8.9 times due to inoculation of microbial consortium in sandy loam soil. Guar-gum nanobentonite WDG formulation were found to be best formulation. OL or pH fortification and microbial inoculation brought out changes in microbial diversity, and the abundance of taxonomic and functional gene copies reveal the active microbial guilds involved in the remedial process. Bacterial consortium showed higher degradation of total petroleum hydrocarbon, and half-life were reduced by 2.86 times in naturally contaminated soil. The presence of diverse microbial groups, metabolic activities, and enzymatic activity indicating that added bacterial consortium played a significant role. The developed formulation can be applied at contaminated site, and restoration of soil microbial diversity and functional activities can be achieved. In long term, mass production of microbial culture and scale-up of developed bioformulation in the form of technology and its commercialization will be carried out. To extend the work further, proposal is also submitted to DST for possible consideration. In addition to research work, I have been conferred with DST Fellowship, Innovative Article, Best Article, Student of the Year, and selected for Best PhD Thesis Award. I have been awarded with first position in poster presentation. I was selected for attending 24 days 9 credit course at Israel and secured 99 grade with A+ distinction. Being a national president of ISA, I served in different capacity as chairperson, co-chairperson, and organizing secretary, and got an opportunity to sign MOU with national and international organizations for promoting youth involvement in agriculture and light sector. In addition, I served as editor-in-chief and involved as editor for compilation of souvenir, cabinet, and proceeding of the events. I sincerely acknowledge all the persons who were directly and indirectly involved during my PhD research work. Thank you.
So I would first like to invite Dr. Bansal for the interaction with the student, please.
Okay, thank you. Mr. Ashish?
Yes, sir.
Very good presentation, and I think good piece of work with a very practical significance and value of the work for agriculture and for the environment as a whole. But I have certain questions, like you, of course, mentioned about the bacterial consortia, also, 3 different fungal species, etc. you identified and you talked about it. So my question is, what were the environmental conditions? Did you vary the environmental conditions? When you were doing all these experiments, under what conditions did you do? Did you have more than one environment, for example, temperature conditions or the moisture conditions in the soil, etc.? Did you simulate those in your experiments?
Sir, we have done this experiment under the BOD, particularly in the soil. And first we have taken the IRI soil for the fortification along with oil and the microbe. We have inoculated in that and we identified the best one. And in that we have kept the 27 degrees Celsius temperature and 65% relative humidity. But and also we maintained the 15% field capacity during the incubation.
These are the conditions you applied, right?
Yeah.
But my question is, what will happen if you vary the conditions, which are, otherwise in the real environment and the field conditions changing, with different seasons, for different crops. So therefore, did you try to simulate some of those other than what you have done? Probably not.
Sir, we have isolated the Assam's naturally oil contaminated soil, which was occurred in during 2020. So we took around 2 kg soil from there, and we incubated our isolated best bacterial consortium and fungus into that soil. But obviously, sir, for field condition, it requires the more upscaling of the microbial formulations. So to extend this work further, we submitted a project in the DST so that we can upscale our microbial.
Okay. We have to save time as well. Okay, coming down to the second question. There are already some known bacteria and the microbial consortia, for degrading the kind of products you're talking about it, including crude oil. I'm sure you know about it. So did you use any of those consortia, as a control, particularly for the purpose? They may not be exactly for what you were looking for, but even then, whatever is already known, did you use some of those consortia, as a control in your experiment? How your consortia, whatever the microbial, species you identified, are they different than those? How do you know this?
Sir, we have not used the controlled anyone, but as per the available literature and the technology, one technology is oil Jaffer, that is by the Terry, and
Yes.
they use the consortium in the field condition also, and they are doing from the last 15 years. So
no, that's okay. Don't go into their detail, but did you use any of those?
No, sir, we did not use, sir.
Okay, third question is, you have gone to the level, which is good, identification of some functional genes, and you talked about the enzyme activities. So you think, what are those specific genes in this consortia, in the individual microbial strain or in the consortia collectively, which would have done this job at the enzymatic level? What are those enzymes? Or if you know their functional genes, and contributed by which particular microbial strain in the consortia?
Sir, itself, it is a very, sir, tedious task. But what we have done in general, when the pollutant concentration increasing in the soil, so nitrogen regulation activity is affected. So by seeing that activity, so we have taken in the consideration NIFH, NIRK, and MOA, and NIRS and NMX kind of functional gene. So by looking into that functional gene, and comparing the degradation capability of our microbial consortia, we can say that yes, our microbes are in a better position to degrade the particular environmental contaminants in a degradation in a contaminated soil.
Okay, now quickly, I get reminded of another point here for clarification is this, what is the efficiency of your microbial, consortia? What efficiency? Because you like to increase the efficiency of degradation of the particular compound you're talking about it. So if you want to increase the efficiency of the already identified consortia or microbial strains, how will you do it?
Sir, in our case, we found that, sir, whatever is available, like oil Jaffer, if we talk about, so they take the 4 months time to degrade the oil content if it is up to 14%. And our Assam natural contaminated soil is the 25% oil content, and we took the around 2 months time in the incubation condition for degrading this oil. So we may say that it can also work in the field condition, but we need to see that thing.
Well, if you want to increase the efficiency, how will you do it? That was my next, clarification.
Sir, we can increase the efficiency by biostimulation, as we can also add the ammonium sulfate or some potassium nitrate nutrient in the as a nutrient mixture, and we can also reduce the CN ratio of the particular environment, so the microbial capacity will be increased.
Okay, thank you.
Thank you, sir.
Thank you.
So
sir.
now I would invite Dr. Patra for the interaction purpose.
Yes, thank you, Mr. Assis. Excellent presentation and very much, pertinent, important information you have generated, which is very much relevant for remediation of the contaminations occurring due to the spills over by the petroleum products. Congratulations to you and your chairman and the team for these wonderful studies. Most of my queries, Professor Bansals have asked, but just what I was that the nutrients, when you add the nutrients, there is no impact about the enhancement of the degradation, but the nitrogen cycling microorganisms, their populations, or perhaps the diversity is increasing. That is what you reported. But when you add, the ammonium sulfate, there was hardly any changes about the degradations. And also, when you added the compost, also there is no changes in their performance. But the abundance of the genes, that means the diversity of these nitrifying organisms, even the nitrogen fixers, NIF-H genes, you have seen the more in those contaminated sites. Why these kind of the contradictory observations? Why? Because these nitrifying organisms, when they get substrate, normally their populations and activity increases. But when you are adding the ammonia, your organic manures, or the nitrogen fertilizers, their activity is not means the performance is not increasing. What may be the reasons?
Sir, we have identified this functional genes abundance in the soil where we have not added any nitrogen additional source. So we found that this NIF-H gene, there may be the case that NIF-H is particularly for the nitrogenous enzyme activity. So the atmospheric nitrogen is converted into ammonia. So there may be the case that when soil is in a condition where high carbon is there, but our added inoculum that is helping to capture that particular nitrogen, or maybe there is the case that they are increasing the population of some other bacteria that are present in that environment. So they are taking the atmospheric nitrogen, converting that nitrogen into the ammonia for their use and for their rapid growth and for more population increasing. So that's why the degradation capacity is also enhancing in that particular environment.
Okay, thank you.
May we now have the comments from Dr. Patil, please?
Mr. Ashish, a very good presentation you have made. Just I have two questions. Number one, how much oil contaminated soil in India? And secondly, as can the same consortia be used for oil spill over the ocean, because oil spill in ocean is a big problem nowadays? And the same consortia can be used for the remediation? And secondly, or you can have an experiment in a lab also, taking water and then you spill the oil and you take the experiments. The same consortia will be applicable, or it will do that work? These are my two questions.
Okay, sir. Sir, as per literature available, around 20 to 25,000 hectare area is contaminated with crude oil during the last 20 years in India. And the thing is that we have also seen the efficacy of this bacterial consortium in the risk minimal medium. So if we compare the medium and we can compare the ocean, so by that way we can say that our bacterial consortium is also best for degrading in the medium. So it may also work in the ocean also. But yes, sir, obviously we need to do the experiment with ocean water also.
Okay.
Okay, thank you, sir. So may now we have the comments from the chairperson, sir.
Thank you, Dr. Sir. Thank you very much. Mr. Khandalwal or Dr. Khandalwal. I hope Viva is over. Congratulations for very good work, very good presentation, very relevant, pertinent topic for the project. So I was really worried what Dr. Bansal asked. Always we have something to build upon. Only rarely we come across something where we work de novo. Science has progressed so much. So any reason you didn't have the control because we want to make progress, do something better than whatever is available already. But definitely because you have published papers in a very good journals, so there must be some solid reason for acceptance of the papers.
Sir,
Because
yes, sir.
if we make some progress only, that will be accepted for publication.
Sir, for control, what we have done, we have contaminated the soil with the oil, and how much oil is degraded by the natural biota that is present in the soil. We have taken that as a control. But yes, sir.
So you see, the work on ocean is going on since 1970s. One of my Korean colleagues worked on bacteria, and then came reports about that. So there must be something already, or if there is no, but your quality of work must be very good. That's why it has been accepted. So I wanted to know what was the good point in spite of that major limitation.
Sir, we tried to purchase some, we tried to contact with some oil industries also, and also with the already available product that is available in the market, but they provide material only to the contaminated sites to treat that particular material. They don't provide for the experimental purpose. So that's why we have not taken any control, whatever technology.
You mean they are not interested in any improvement?
Sir,
or they have some sort of patent?
Yes, sir, they have patented. Oil Jaffer is the patented technology.
But still they must be giving it to distributing to the farmers.
Sir, they take the project and they do remediation from their own. Hello.
Those very soils. Yeah, just for future studies. It's more for the faculty rather than you. But I come to similar question. When you say extraction methods, did you compare extraction method also with the standard ones, or you went with new extraction methods?
Yes, sir. Sir, some EPA guidelines are there. So we have taken that method is also exent DCM we have taken is a 1:1. So
okay, fine. You have answered my question that you have guidelines you have followed. You said that the consortium which you have works under different conditions. Just a stray point came to my mind. Will it work in paddy straw?
No, sir, we did not try, sir.
Or maybe other colleagues who are working, because you also mentioned that there are different diversity of microbes in different soils, etc. So it means it varies deep. There are different microbes under different situations, but at the same time, you said that it works under different conditions, even may work in ocean also. So maybe worth trying, because if someone can eat wood, maybe it can eat wood everywhere. Very good example is termite. It just eats everything. Then I was just, again, this is more personal, but I, as an old man, and I feel my duty to tell what I feel. Maybe it may be helpful, it may not be helpful. I am not a god, I am a human being. When I went to South India, I was in ICR for a long time, 8 years. And my host always tried to carry me to some hotel or restaurant where I can get roti, dal roti, rather than only chawal. So हम वही परोसते हैं जो आगे चाहिए किसी को. Do you think you are being president of ASA is a plus point in such presentation when there are, excuse me, when there are scientists in the jury? It may be a very good point if you want to have ticket for MLA MP. Why I am saying this, that I have got many invitations as Vice Chancellor from ISA. I all the time politely refuse to participate. So please see the reader when you are writing a paper in view, or see the jury or the team who are going to review in view. I assure you that it will not have any adverse effect, but for your future. Present from the point of view for which you are going to examine it, or supporting data. Anyway, thank you. Thank you very much.
Thank you, sir. So it seems Director sir would like to interact with the student. So I invite him.
Sorry for coming after the chairperson.
No, it's okay. Okay, no problem.
It's just this afterthought, one of the classical work done on the, multiplasmid hydrocarbon degrading bacteria by Anand Mohan Chakraborty, the Pseudomonas. In 1983, I can remember International Genetics Congress in India that was held. He presented his work, and this was a national news for many, many days. So when the question were being asked on the control, at least if you don't have access to that Pseudomonas which was patented, did you compare the Pseudomonas multiplasmid strains from within India as a control in your experiment, or can you comment on that?
Yes, sir. Sir, we have Pseudomonas, two species of Pseudomonas that we isolated from the contaminated soil. One is the Pseudomonas erythrae and other is the Pseudomonas aeruginosa. And Pseudomonas aeruginosa also gave very good result, but it has the it is pathogenic in the nature. So we did not take this Pseudomonas aeruginosa for further consideration of the bacterial consortium preparation. So we have taken Pseudomonas erythrae and Bacillus amyloliquefaciens, because Bacillus amyloliquefaciens, what he does, we have the C11 to C36 hydrocarbon. So some are lower molecular weight hydrocarbon, some are more molecular weight hydrocarbon. So this Bacillus amyloliquefaciens basically works on the higher molecular range.
It's okay. I got the answer. My question was that, when this was being asked, that should have been your, response, that you have taken Pseudomonas and you should have known Pseudomonas has been genetically engineered, used, patented, and as the most important technology by Anand Mohan Chakraborty those days. Yeah.
Thank you, sir. So with this, now we'll move on to the next presentation, which is by Ms. Nityeshwari from Discipline of Agriculture Economics.
Good morning, everyone. I am Nityeshwari, presenting significant research accomplishment of my PhD program. During the developmental transition, the country needs to create more value addition to the commodities produced from the primary sector. With the sharp decrease in the number of workers in agriculture, women workers are expected to grow faster and absorb surplus labor in the country. Increasing demand for processed food due to rise in per capita income and changing consumption pattern led to more demand for processed food. More importantly, reorientation of the global trade towards high value commodities perceived as the most beneficial to the growth in the country. Realizing the potential, the food processing sector is considered as sunrise sector in India. Various policy initiatives by the Indian government during the 1970s and 80s reveals the nation effort to bring the economy into the forefront. Country has set up a separate ministry dedicated to food processing in 1988. Several other initiatives have been taken thereafter. The most prominent ones are the approval of 100% FDI through automatic route and Pradhan Mantri Kisan Sampada Yojana. Despite this, level of processing in India is very low and often reported as sluggish and lacks competitiveness, and ranks 17th in the processed food export globally. On the other side, there exists a significant amount of wastage due to harvest and post-harvest losses, and the trend is rising. Price volatility, distress sale are the other concerns. Finally, the imbalance of production and utilization. In this context, past studies suggested that processing sector in the country is mainly constrained by stagnant technical change and limited resource utilization, but there is a scarcity in understanding the driving factors associated with it. To fill this research gap, the study was proposed on investment and development of food processing industry, firm level evidence from India, with the following objectives: to study the structure and regulation of food processing industry, to analyze the pattern of factor intensity, resource use efficiency and productivity, to assess the pattern of investment and role of FDI on productivity, and to examine policy and institutional imperatives for the development of food processing industry. To accomplish these objectives, the study used secondary data obtained from various sources, mainly Annual Survey of Industry, DIPP, Ministry of Commerce and Industry, Ministry of Corporate Affairs, CMIE and others. Food processing industry in India is characterized by small units, of which 56% are present in rural area. Investment in research and development and foreign direct investment is meager. Only 0.84% of the firms have R&D units and 1.09% of the firms received a foreign capital share. The distribution of firm across the scale of operation showed most of the firms are micro, small and medium in size, and hardly 2% of the firms are having a large size with the turnover more than rupees 250 crores. The industry output in constant term is estimated to be 8,150 billion rupees, of which traditional sectors like grain, starch, dairy, oils and fats and sugar industry contributes 68%. During 1980 to 2017, the industry output growth rate was recorded 7.59%, but during this period, the temporal trend in the ratio of gross value added to output remains same. This reveals that for producing output worth of 100, almost 90 rupees has to be spent on the purchase of material. Across the subsector, most of the commodities indicated similar kind of ratio. Exceptionally, processed fruits and vegetables, bakery, sugar, confectionery and prepared meals and dishes reached a higher ratio of around 20%. The potential of employment generation in food processing industry was examined using employment function. Results reiterate the positive effect on employment. 2.8% increase in output will generate 1% additional employment. This effect is more promising in high value commodities such as meat, fish and feed industries. However, the profit rate and wage rate of food industry found to be low as compared with the industry average, which may come in the way of attracting and retaining skilled workers in this industry. Industrial productivity are significant source of national income and they are root to standard of living and global competitiveness. Productivity performance of food processing industry was carried out by considering two major components of productivity, that is efficiency and technical change. Estimation of resource use efficiency was carried out using semi-parametric four-stage DEA framework, which combines frequently used DEA and stochastic frontier analysis. The efficiency score of FPI during 2013 to 2018 was found to be low, that is 11%. Inefficiency of resource use is associated with the capital use, which is visible in terms of mean inputs, lack of fixed capital, building and plant and machinery. The effect of macro environment on firms' resource use reveals regional policy and business environment across the states have a significant impact. Import intensity that enables more openness and investment intensity in plant and machinery facilitates in reducing input slacks and improve the resource use efficiency. At the firm level, an increase in firm's quick ratio by 1 standard deviation will increase efficiency by 7%. Timely and quality raw material play a significant role to improve resource use efficiency. For analyzing total factor productivity, the study adopted the control function approach. For robustness check, two alternate methods were used. The analysis clearly shows that growth of processing industry is largely driven by the combined input growth, that is 3.91% and TFP by 2.73%. The technological change and the gain was more significant only after 2004 and 5. But advances in technology has not translated in reducing real price of processed food, wherein its growth rate was 8.64%. FDI spillover is more prominent and considered as the starting point of modern technology transfer. However, the possibility of crowding effect of domestic investment without generating technological benefits may also possible. There are different sources of spillovers. They are horizontal spillover through competition and demonstration, vertical spillover via background and forward linkages, and through human capital by migration and ownership. FDI inflow in food processing industry indicated increasing trend with a significant hike from 2011-12 onwards. Across the product groups, feed industry attracted the highest investment, followed by oils and fats, and miscellaneous products includes ready to eat and cook foods. The productivity spillover effect in FPI was analyzed by the random effect panel data regression model with TFP as dependent variable. The results indicated a positive relationship between the productivity and FDI spillovers with a significant effect within industry. There is also an indication of positive vertical spillover via backward linkages. The extended model disaggregates the spillover effect by the country of origin, showed the prominent effect of spillover from the developing country source of origin. Similarly, significant effect of spillover was found in FDI that enters through automatic route. With this, we found that growth of FPI was largely driven by the technical change. Resource use efficiency of FPI is very low. Incremental output will generate additional employment. With the presence of foreign affiliates, there is rise in domestic productivity. Having understanding processing industry scenario, in India, we concluded that food processing industry in India has begun to expand but not yet realized its potential. The differential effect of productivity spillover showed absorptive capacity of the domestic firms in the food industry has not been sufficient to generate the spillover gain from the technical pioneer. For optimizing the resource use, macro level regional policy and business environment, innovation in the industry has a significant impact, but R&D intensity of the country in FPI is negligible. Therefore, incentives for investing in R&D is the need of the hour. Encouraging institutional support and policies addressing the above issues will enhance the development of food processing industry. Further, nature of technology and their adaptability by the domestic firms can be explored. Consumer preference and willingness to pay for the processed food and policy instruments supporting quality attributes can be studied. From this work, one paper is published with the NAS rating 5.30 and another paper is accepted with the NAS rating 5.89. One more article is accepted with the NAS rating 10.48, and we have also published one book chapter and popular article. This work is presented in different national and international conferences in the form of paper and poster presentation. During the PhD, I received research grant under Uma Lile Mentorship Award. As a part of co-curriculum activity, I secured second prize in national level essay competition. At the end, I would like to thank my chairperson and advisory committee, Director IARI, PG School IARI, Head, Professor, esteemed colleagues and students of the Division of Agricultural Economics, IARI New Delhi, and friends and my family members. Thank you.
Yeah, I would like to first invite Professor K. Singh for the interaction, please.
Thank you, Dr. Dauja. So, Nitya Shri.
Yes, sir.
Okay. A very good work on food processing sector. When we talk about food processing, as you indicated also that efforts are being made since 80s, still the achievement is not up to the mark. So do you think that there is a direct relationship between the marketable surplus and the food processing sector? When you presented the coefficient, it showed that there is more possibility in fish, fruits and vegetables and high value crops for employment generation through food processing. So did you try for sectoral analysis? That is, what is the status of food processing in fruits and vegetables or fish sector or animal sector and so on?
Yes, sir. We did the similar kind of analysis for subsector also individually, because these sectors are promising because they have started to begin right now. So the progress can be made very fast. That is the one reason, sir. And the domestic part you are telling that also been tested with the linear regression model, and it is suggesting that when domestic market is progressing more, that is having more impact on the value addition.
What is your inference on the basis of the import intensity, which you have worked out?
Sir, import intensity, the variable which we chosen as a ratio of the imported to the output, import intensity is coming in the third preference, sir, as the macro variables when we listed. Though it is significant, first it is coming the technology intensity, followed by the state regional progress, then the technology import intensity. It is having significance, but in the order of preference, it is going the last one, sir.
Can you suggest one policy which will help the government to boost the food processing sector on the basis of your study?
Sir, to give the first choice, it is the consolidation and aggregation of the product. It is very important, sir, in the primary sector where it is very fragmented. Once it is done, I think it is helpful for the processors to pick up the product very easily. So that channelization will help to a great extent.
Okay, thank you. Thank you, Dr.
Thank you, sir. May I now invite Dr. Bansal for the interaction, please?
Okay, thank you. Ms. Nityashri, it's a good presentation. Of course, I must admit that economics, I hardly understand anything, technically what you mentioned. But we all have interest in food processing industry in India. And I got some of the answer of the question which I thought I would ask you from what Professor R.K. Singh asked you. And I think this industry, despite the fact that the government is supporting it so much, even there is a ministry, which we never had before, Ministry of Food Processing, and still it's not, coming up in India at all. India is a diverse country. I'm sure you know about it. So when we talk of food processing, which particular sector, I don't know, subsector, that's what probably Professor R.K. Singh mentioned. We're talking of cereals food processing, we're talking of fruits, we're talking of, we have got so many. Otherwise, I come from plant genetic resources. There are so many, nutrient-rich, foods, which could be processed. So which particular sector are we thinking about it? And what is your answer, general, for the awareness point of view, that why this particular food processing industry is not coming in India? It is something to do with the consumers, it's something to do with the mindset of the people as well in India, the kind of education we have or the preference we have for the food processing. So I think overall as a PhD student, what do you think your response would be?
Sir, India, we have treated food processing industry as a traditional sector, first of all. So that idea should be changed to the sophistication of the industry, because now, to give an example, like we are talking about the Amul kind of a thing, which is doing well in a country. But when we see this comparison to the international domain, the Switzerland is having already 100 of varieties of cheese, but we have only very 10 or 20 varieties. So market is so specific and consumer is demanding so much. I think that much supply is not coming from the Indian market side, sir. So that is the one.
Okay, anyway, quickly also like to ask one of your conclusions was that we would like to have more investment in R&D.
Yes, sir.
So what kind of R&D, particularly with regard to food processing industry, you're talking about?
Sir, one could be the processing machinery, we are having a lot of constraints, sir. A lot of machineries are coming from the overseas, and some majority are the patented ones also. This will cost more for the small size sectors, what we have in the Indian context, to adopt those technologies. If it is made innovative way to available to these small firms, it will be very useful for them to come up with the
So do you think whatever the products we are producing as as agricultural scientists, like for example, different varieties in whether in cereals or fruits and vegetables, are they suitable enough for processing?
Sir, not many, sir. Most of the time, it is also a concern that all the pulses cannot be suitable for particular level of processing, and all the potatoes are not suitable for processing of a kind of a chips. So that is also a major concern, sir.
Okay, thank you.
Thank you, sir.
Quick suggestion. Your presentation, when you were recording, volume should have been taken care of very well. Sometimes you want to convey a message, you have to see whether it's being conveyed properly to the audience or not. I think for future, you need an improvement there.
Sure, sir.
Thank you.
Thank you, sir. So can we have the questions or queries from Dr. Patra, please?
Yeah, thank you. Dr. Nityasri, I understand your Vivac examination has been, it is done, and now you are doctor. So hearty congratulations, excellent presentation in the context of national economy. This is very, very important and necessary for the country. So in that context, very much relevant your work. My questions may not be, directly what you have done, but some information you may, I think, give to me. And this is, in 90s, with economic liberalization, I remember, because I was at the Central Institute of Fisheries Education, Mumbai. That time, many companies, food processing industry in 90s on seafoods, the different aquatic foods and other things, they established with public equity participations. And then with time, they are all, I think, closed. In that, there was foreign affiliations also, foreign participations also were there. So in one hand, maybe, this kind of things happen, but then sustainability in a long run, maybe economic viability and other things were very, very poor, or maybe some other reasons. So those are not seen nowadays. What may be the reasons, and which is also very much relevant in the context of your work?
Sir, this kind of incidents are common, sir. To give another example, in Bihar, lychee beverage industry also started and closed. The main reason they quoted is that the environment, the surrounding, the power supply, the road one, we call it as an infrastructure facility. This has to be complement that investment what private people are doing, either from domestic or overseas. If that is not complementing, the establishment will not sustain and it will not become economically viable, sir. That is the possible reason.
Okay, thank you. All the best.
Thank you, sir.
Is it my turn, Dr. Dhuja?
May we have the comments from Dr. Patil, sir?
Okay, Dr. Patil is there. Yeah, please. Sorry, I'm sorry.
Presentation. You know that Government of India has set up or established separate Ministry of Food Processing. Still, our sector is not growing. Is the farmer produce organization is a solution for that? Because some of the farmer producer organizations, like in Maharashtra, Sahyadri Farms, is actually doing excellently well. The turnover actually increased up to almost 700 to 800 crore rupees. So whether the farmer producer organization is a solution for this boosting this sector?
Sir, it is a promising policy initiative. It has been taken as an institutional innovation, sir, because we have a cooperative model in our country that has been not successful in many commodities. That's why this farmer producer company came into picture. Again, for this farmer producer companies interacting with the BODs and CEOs, the main starting like the duration, what we call it as a 5 years, sir. If they could able to sustain it, definitely it will become a great solution for the processing sector, sir. In that way, it is a good initiative, sir.
Okay.
Thank you, sir. So I could see Dr. Abhijeetkar raise the hand. Dr. Abhijeetkar, would you like to ask any question?
Chairman, sir, can I quickly ask one question? If one more.
Can you hear me, sir?
Yeah.
I just wanted to ask Nityashree one question. She had given an example as to the success of Amul and the success of others vis-a-vis the non-success of maybe fruits and vegetable sector. Has she tried to find out as to why this has happened, why this significant difference in the success of maybe Amul or other dairy industries compared to the vegetable industries? What has been the major reasons of their success and the failure of the other food processing industries? That same thing would suffice even for FDI in dairy compared to FDI in other fruits and vegetable industry.
Sir, one reason is though milk is highly perishable as compared to the fruits and vegetables, it can be grouped and channelized, but that is not possible in case of fruits and vegetables, sir. And in Indian market, two kinds of preference still exist. We prefer fresh rather than the processed fruits and vegetables. So this has to be in a different form so that it should be supplied in the market. Otherwise, this sector is not accepting that kind of a processing, sir. And one more thing is that it is coming from the smallholders things. The collection and consolidation and processing having a loss at each stages. If the channel is lengthy and wastage is lengthy, and the margin will be very low. So that's why the investment also a kind of a limitation in this particular sector, sir.
But don't you think that the raw material availability of a given quality and a given variety, of the logistics which you are saying, is a major cause of concern in India, especially in fruits and vegetables?
Yes, sir. It is a major concern, sir. It is having a huge impact in the processing sector, sir.
Okay. Thank you. Thank you, Nityashree.
Thank you. So, Dr. Bansal, would you like to ask?
I think I will, for the sake of time, skip over. We'll have the clarification later.
So we can move on to the comments from the chairperson, sir.
Thank you, Dr. Sir. Thank you very much. First of all, welcome to Dr. Kesing. Dr. Kesing, I'm sorry, I missed you. Then Dr. Nityashree, very good presentation, very important research project. Whenever someone asks me about Punjab or Northwest India or other regions, say coastal regions, then I always say that follow Maharashtra. Processing and export are only avenue, as now food production has increased much beyond our requirement. But before asking any question to you, I will bring some information to your knowledge. You said a policy started in 1970s. Policy initiative started in 1960s. MSP, FCI, credit facilities. I will not go in detail. Credit was visible earlier also, but they took a very important decision in relation to Punjab and Haryana. At that time, Punjab was one. So it's secondly, I really feel I don't know what should I say, that we criticize ourselves too much. You see, when I was student at my alma mater at IRI, and I heard Dr. Swaminathan many times. Besides, he taught me 2 courses in addition to many lectures, otherwise general lectures. He all the time said that CFTRI should be with the ICR. So I said, did not have this allocation or duty. Let CSIR be criticized for that. ICR started CFT only in, I think, 80s. I don't remember the exact year. I will see that. CFT was established much later. Then where was raw material to process. We start comparing with other countries. When did we have some reasonable exportable surplus or something? And with the rice becoming important, rice milling industry came into existence. So this balanced view, I think, yeah. Then directly proportion, more the value and more volatility. And 88% of farmers, latest data I read somewhere that 88% are small and margin farmers. So they cannot afford to have the crops volatility, where there is high volatility. Then in case of a question asked by one of the colleagues, you give very good answer. You are very, very correct. But another factor is Korean, the person. Read his autobiography. I don't want to repeat some sentences from that. And now coming to, I will want to get knowledge from you, not questioning you. With the population size and with farmers being small and marginal, and collection being one major factor, and machinery also, as you said, we need to adapt. We cannot introduce machinery as such. Should we focus only on MSME, or we should? Dr. Vansu, you said you don't know much about economics. After becoming vice chancellor, I feel I should have gone to economics rather than plant-breeding genetics. They dominate. But just adopt they just introduce models from USA. I want to adopt in India. Anyway, Dr. Niteshri, if you can just guide me, because this question is posed to me.
Sir, regarding MSME, sir.
MSME, having part-time farmers, because we are too much tied to land also, at least Punjab, Haryana, Western UP. Even if we have one canal, we say, oh, I am a farmer. I am Jimidar.
Sir, MSME can be taken in a different manner, sir. Like, because recently they have revised the criteria. Upward revision.
No, leave aside government policy. As a scientist, should we lay more emphasis on MSME in this food processing?
Sir, yes, sir. Because major part of the firms will go to MSME only.
Okay. Then another, I will say, question, or again, I want to get knowledge or information from you. Which area should we focus on? You said animal feed. Animal feed, yes, you are right. If we have good quality animal feed, all maize produced in Punjab and Haryana can be used. But when we think in terms of export, which area? You see, the raw material is not always very, very important. If you have to produce a juice, normally second quality fruit is taken. But if you have to process like potato fry, quality of raw material is not always relevant, depend on the product what we want to have. So which area we should emphasize when we want to process and export?
Sir, leaving the grain apart, because we are dominated in the grain industry, the second preference should be given for fruits and vegetables, sir, because these days plant-based
fruit. I don't like this term fruit and vegetable pulses. I'll see it. Mention the fruit, sir, or the vegetable.
Sir, now for fruit, I would say lychee is the one we should.
Oh, lychee is only small niche in Himachal Pradesh and small niche in Punjab.
And banana, guava is another thing, I would say, sir, for fruits.
Guava juice, banana juice? Banana juice?
Not only juice, sir, even bars can be prepared. They have long shelf life rather than juice. They are much better than the chocolates what we eat. So,
technologists also in the audience. My impression that we are working more on in research and development. I agree with you. More money should come. We are working more on recipes rather than. I'm talking about PAU. More on recipes rather than technologies. Anyway, thank you, ma'am. Thank you, Niteshri. Wonderful. Pleasure hearing you.
Thank you, sir. So let's move on to the next presentation, which is by Ms. Sangeeta Bhattacharya from Discipline of Agriculture Extension.
Good morning, respected jury members and my dear friends. I am Sangeeta Bhattacharya, representing the discipline of Agricultural Extension. I welcome you all to my presentation. Let me begin my presentation with a question. How many of us are aware that rural India, housing 68% of India's population, have access to only 25% of India's healthcare infrastructure? Less than 80% of rural households have access to piped water facilities. Why are news articles questioning the status of poor educational facilities in India, and why there exists a wide urban and rural disparity with respect to access to sanitation facilities in India? All these news articles reminding us time and again that rural development is the need of the hour. Considering the words of Mahatma that the soul of India lives in its villages, Government of India has established a Ministry of Rural Development, which has the third highest ministry-wise budgetary allocation and rolls out several schemes of rural development every year. But most important is to evaluate the ground level impact of such schemes. One such ambitious rural development program named Sansad Adarsh Gram Yojana or SAGI was launched in 2014, in which each member of parliament was to adopt and develop villages into model villages by 2024. Till date, more than 2,000 Gram Panchayats have been covered under this program. But there exist certain villages in India like Hivra Bazar in Maharashtra, Ganga Devipalli in Telangana, Pansari in Gujarat, and Ramachandrapuram in Telangana, and few more other villages which have achieved the epitomes of development through the individual efforts of philanthropists and have been declared by Ministry of Human Resource Development as model villages even before the launch of the SAGI program. So we see that not only government programs like SAGI, but also simultaneously different development efforts are going on to develop villages into model villages. But the question is, what are the indicators of a model village and how can we measure the progressiveness of a village? Is there any robust methodology for that? So the ultimate need is to quantify and standardize indicators for measuring the rural progressiveness for rural development to actually happen at grassroots levels in India. This brings us to the obvious research gaps that the current status of development of SAGI villages has not yet been compared with the model villages developed by individual efforts. There is a lack of common indices for evaluating such villages. Socioeconomic impact of SAGI has not yet been documented at village level, and there is lack of conclusive suggestion on ways of strengthening SAGI. Hence, the study titled Socioeconomic Impact of Sansad Adarsh Gram Yojana in Maharashtra and Telangana was formulated. Study had 5 objectives. First, to delineate the structural and functional mechanism of SAGI. Second, to identify and standardize indicators of model village and assess the performance of villages adopted under SAGI. Third, to analyze the perceived facilitating and inhibiting factors in developing a model village. Fourth, to study the socioeconomic impact of SAGI. And fifth, to design suitable strategies for convergence. The study was purposefully conducted in the states of Maharashtra and Telangana with 2 model and 2 SAGI villages selected from each states. The states were selected purposefully because these had the maximum concentration of model villages developed by individual efforts, and to facilitate better comparison with SAGI villages, these states were selected. Mixed models were used of different research designs. In Objective 1, the structural and functional mechanism of SAGI was studied. Structural mechanism of SAGI revealed that it was headed by a national level committee with corresponding state and district level committees. The functional mechanism of SAGI showed that SAGI was being implemented stepwise, starting from identification of Gram Panchayat by Member of Parliament to formulation of village development plan with participation of villagers, and then execution of projects targeting the holistic development of villages. With a sample size of 240, the second objective was executed and 7 rural development indices were developed using NUPA and equal weightage method, index values calculated for each village for assessing their performances. The first index developed was Rural Health Status Index through identification of 8 rural health indicators. Categorical PCA was then done to obtain weightages for each indicator. The Cronbach alpha was seen to be sufficient and percentage of variance was also more than 50%. The weightages for each indicator was obtained and then multiplied with normalized values, summed up and then divided by grand total weight through NUPA method to obtain the final formula for Rural Health Status Index. Several other indicators of various rural development parameters were identified, and similarly 6 other indices were developed using the NUPA method: Rural Educational Status Index, Rural Farming Status Index, Rural Livestock Status Index, Rural Ecological Status Index, and Status of Rural Women Index. The final formulae for the various indices developed are as follows. The 7th index, Rural Infrastructure Index, was developed depicting the various amenities present or absent in the village, and equal weightage method was used for developing this index. The index values after calculation showed that the SAGI villages in Maharashtra excelled in farming and health parameters as compared to the model villages developed by individual efforts. In Telangana, the SAGI villages excelled in education, farming, livestock status and ecological parameters as compared to the individually developed model villages. The perception of 120 respondents of SAGI villages were measured through a multidimensional perception scale in the third objective, and perceived factors identified through exploratory factor analysis. Government public liaison, infrastructure and education, collective power of villagers, local political environment, equality and cultural inherent values of villagers were perceived as facilitating factors in implementation of SAGI, while faulty adoption policy, bureaucratic hurdles, village factions, lack of funding, lack of coordination were perceived as inhibiting factors of SAGI. Individual perception scores calculated through multidimensional perception scale revealed that Tikakarwadi and Dandepalli villages had favorable, Ainovolu villages had neutral, and Maluncha-Budruk villages had unfavorable perception of SAGI, and the scores were significantly different. In fourth objective, a non-SAGI village was identified from each of the state, and with without model was used to study socioeconomic impact of SAGI. Economic impact was measured through differences in occupational status, monthly family income, number of earning members in the family, and material possession status. Social impact was measured through differences in social connectedness, family educational status, social prestige, and satisfaction level. Occupational status results revealed that SAGI villagers of Maharashtra mostly had clerical jobs, while non-SAGI and both categories of villagers of Telangana were self-employed. The Man-Whitney test revealed there was no significant differences in occupational status of SAGI and non-SAGI villagers. In Maharashtra, majority respondents in SAGI and non-SAGI villages had monthly family income of 50 to 75,000, while in Telangana, SAGI villagers had 75,000 to 1 lakh family income as compared to their non-SAGI counterparts. The Man-Whitney test revealed the differences were significant. With respect to number of earning members in the families, majority families of SAGI villages in both states had more number of earning members in their families than their non-SAGI counterparts, and the Man-Whitney test revealed that the significant differences existed. In both states, majority families of SAGI villages had better material possession status than their non-SAGI counterparts, and the differences were significant. All these significant economic impacts can be attributed to the reason that livelihood opportunities increased in the villages after adoption under SAGI program. In social connectedness, SAGI villages had more scores in social participation and mass media exposure, which was reflected in both states through their social connectedness median scores, and the differences were significant. SAGI villages also had better educational status than their non-SAGI counterparts, and the differences were significant. Social prestige scores were also more in families of SAGI villages in both states, but the differences were not significant. Satisfaction level of SAGI villagers were more in both the states, and the results were significant. Significant social impact parameters highlighted that SAGI villagers had faith in the long-term impact of the program. Alpha race and tougher method was used to develop strategies for convergence of state departments in the fifth objective in four broad dimensions of identification of need, planning, technical expertise, and funding with sub-strategies under them, and the most important dimension for strategy making was revealed as convergence required in funding. Coming to the key findings of the study was that Saji villages were faring well in terms of health, farming, livestock, ecological status, education, but at the same time was being plagued by certain inhibiting factors. Saji had significant impact in terms of increase in monthly family income, number of earning members in family, material position status, social connectedness, family educational status, and satisfaction level of villages, but was plagued by lack of funding. Implications of the study where ground level scenario of SAGI was revealed. Indices developed can also be used to measure rural progressiveness in future programs. Multidimensional perception scale can also be used for future rural development programs. The study also pinpointed areas requiring convergence and smoother implementation of SAGI. What is the way forward? Ensuring convergence and dovetailing of schemes, leveraging on facilitating factors, correction of faulty adoption policy of the program, and involvement of non-political bodies. Lastly, coming to my publications, I have published 2 papers in NAS rating above 6 and I have 3 papers in journals NAS rated above 5. I have received Outstanding Research Scholar Award and Best Poster Award in National Seminar and also delivered oral presentation. In extracurricular arena also I have several awards to my name. At the outset, I would like to thank all who have been a part of my PhD journey. Lastly, ending my presentation with the words of Sri Aurobindo, the village cell is the cell of the national body and the cell life must be healthy and developed for the national body to be healthy and developed. Thank you.
So may we have first the comments from Professor Rakesh Singh?
Thank you, Dr. Dahuja. Ms. Sangeeta.
Yes, sir.
Very good work. I have only 2-3 small questions. While studying the structural and operational things, did you find that what was the basis for selection of a village under the SAGI?
Yes, sir. Thank you, sir, for your question. Sir, actually, the basis for selection of villages under SAGI was just the population size of the village. That each member of the parliament has to choose one village from his or her constituency other than the village to which he or she belongs, and the population size should be minimum 3,000 to 5,000 for plain areas, and in hilly areas it was like 1,000 to 3,000. So just on the basis of population size, villages were being selected.
So my suggestion is for this, you are the social scientist, that even since independence, since the policy initiative, we don't have any index on which basis we can identify that, okay, this intervention should be in this village and then that village. Means that is the most critical part, the most critical element which should be developed. Whatever the villages you covered, did it fall in aspirational districts?
No, sir, it was not a part of the aspirational district program, sir.
Okay. While studying the socio-economic impact, you compared the SAGI village with non-SAGI village. Is it?
Yes. Yes, sir.
So, did you also compare that before intervention and after intervention of SAGI?
Actually, I could not do that, sir, because before intervention, the data was not present with me.
Unless until if we assess the impact and we compare before intervention of the SAGI and after intervention of the SAGI, I think mere comparison between non-SAGI and SAGI will not give the true picture of the socio-economic, of this economic impact of the any intervention. So that,
yes,
that we should keep in mind. Thank you very much. Thank you.
Thank you, sir. May I now invite Dr. Bansal for the questions or queries?
Ms. Sangeeta,
Yes,
see,
sir.
you talked about the Ministry of Rural Development, right?
Yes, sir.
And you gave all that background. Of course, this is not my area of expertise, I must admit that again. And but we work under the Ministry of Agriculture, right? Which is also Ministry of Agriculture and Farmers Welfare.
Yes.
So my question is, while you do all that, what you have done, which is fine. And but the question is, for my knowledge point of view as well, that are we not able to look a comprehensive point of view, including agriculture as a part of it? Because when you talk of a rural development, I don't think, this is my limited knowledge, that anything without agriculture being a part of it, whatever model you develop it, the kind of terminology, extension, or in economics for that matter. But for the common person to understand, the developmental part of a village could be without agriculture as well. Do you think so? So if my question makes sense, then what is your say about this, please?
Thank you, sir, for your question. As you rightly pointed out, that without agriculture, rural development is totally impossible. So that is why I had kept one index in my study, that status of rural farming, for rural farming status index I had developed and measured also the farming parameters of the selected villages. And like even in the SAGI program, which I have mentioned, they have also set one objective that means in order to develop the farming components of the village. So a model village or rural development without agriculture cannot happen, means cannot happen in isolation. So now government is also thinking about it, and they have kept one parameter related to farming. So in which I have, when I visited the villages, I saw that the state department officials, like agricultural personnel who are working there, they visit the villages means once in a month, or like some in a frequency like that, so that technologies are covered. And even, sir, I think every one of us knows that already our agriculture institutes are running model village program in their own capacities. Like IRI has their own model village, NDRI has their own model village.
Okay,
so,
fine. No, we should keep the answer, in fact, also a little more focused and within words. Also, my question is for, again, once you have a rural development, you presented your work. On the other hand, we have got, extension related issues and problems, directly of a technology or a seed or fertilizer, whatever we talk about it, directly to the farmer's field, right? So out of the two, which one is to be more preferred, in fact, particularly looking into the current Indian agriculture scenario? We should go into the rural development, this model village concept, or we should go more into the extension agriculture, especially, with regard to the equipment or any other development, talking about, let's say, an IRI to the field for agriculture, for farmers.
Sir, an integrated approach would be more means helpful for the villagers. I found that even in when they were receiving inputs from the extension personnel, but there were other different developmental issues, hurdles in the village. Like there was no proper roads in the village. I mean, one of the village in which I visited, Malunja Budruk, they means in rainy season nobody can venture inside the village. So even the personnel, extension personnel or scientist or KVK or other development department personnel, means due to lack of connectivity, they cannot enter inside the village to conduct demonstrations. So I think, sir, an integrated approach would be more helpful in such cases.
Okay, thank you. Thank you.
Thank you, sir.
Thank you, sir. So can we have the questions from Dr. Patra?
Yeah, thank you. Dr. Sangeeta, excellent presentation. I should say my just simple questions, and these are, the intervention of SAGI, and lot of things happen, and most are positive things. That is very much in your presentation, and that is also obviously expected. In this context, one important is, the natural resource, use, conservation and sustainable use, particularly the water, soil, and the biodiversity. In your indicators, these things, have you included these things? Because in a long run, what I see in the villages, that the ecosystem of the villages is degrading, and with the development, lot of pollutions is also happening. So have you included the natural resource sustainable use, its conservations and based management? This is one. Another thing is that, these villages, Sagi villages, in the context also, Professor Rakesh Singh ji also, prior to Sagi, maybe there are many other organizations working now apart from Sagi, to uplift man to, because of the more attentions since the MP is involved in these model villages, and the funding is involved and all that. So maybe the other organizations, so the resources, the financial resources, lot of many trainings and other things apart from Sagi is also happening in those villages which you adopted in your studies. Just these two queries or clarifications, please give.
Thank you, sir, for your question. First question I would like to answer, sir. I have included these indicators in my study. One index I have, means out of the 7 indices I developed, one was Rural Ecological Status Index, in which, as you mentioned, that sustainable use of natural resources, I had included certain indicators like whether there is presence of water bodies in the village and how these water bodies were being used, whether they were clean or polluted or contaminated, and what are the like whether they are using plastics for their day-to-day usage. So certain indicators related to ecology I had kept, in which presence of water harvesting structure, etc., were also there. In total, there were, sir, like this 8 indicators. Due to the lack of paucity of time, I couldn't present the details of all indicators. And sir, second question, as you said, that already before launch of SAGI, different programs have already been run and individual efforts were also there. So certainly, as I have compared the SAGI villages along with the villages developed by individual efforts, there are also other villages which maybe I have means due to lack of time in a PhD study and all, I couldn't include. But yes, sir, there are certain other villages which are doing their means in their own capacities. They are developing themselves, and under different development agencies, NGOs are working towards this concept of model village.
Okay, thank you. All the best.
Thank you, sir. Thank you.
Thank you, sir. So may we have the comments from the chairperson, sir?
Yeah, thank you, Dr. Duja. Thank you. Dr. Bhattacharya, thank you very much for very important work and very well presented. I'm very happy that you have publication in impact fact journals, which it is not easy in social sciences. And I'm also happy that you work in 2 states. Normally, the tendency is to work in home state. Did you find some difference between 2 states?
Yes, sir. Sir, in Maharashtra, actually, I found that SAGI villages were faring well, means they were performing well in terms of health infrastructure and education. And health infrastructure and farming. And in Telangana, the villages were performing well in terms of 4 other indicators like education, farming, and also health parameters and ecological parameters also. So in like Maharashtra, the focus was a little bit different, and in Telangana, it was differences were found, sir, in both states.
And does it mean that what Dr. Bansal said is true, that the thing in focus should be agriculture and natural resources when we talk of rural development?
Yes, sir. Agriculture is obviously important and it should be in focus, sir. Along with that, I feel that other indicators should also be, means if there is a kind of convergence between agriculture departments.
Dr. Vansal, it is that in Punjab now a different minister for agriculture, different for horticulture, different for soil conservation, different for animal husbandry. How we are talking integrated development, I don't understand. Anyway, let us leave that. The next question is just extension of what agriculture versus rural development, agriculture and rural. Which agro-technology was most pronounced and which was most important but didn't pick up?
Yes, sir. So you're asking agriculture technology, which
Technology
was
most pronounced in terms of effect and otherwise very important but didn't pick up. For example, in Punjab, varieties are adopted, but nobody want to manage a paddy straw.
yes, sir. Sir, agriculture technology pronounced.
No, not agriculture. Just all things which you studied. Everything you studied, other things also.
Sir, like whatever the departments were recommending, they were taking up, and more pronounced, which I found that some kind of infrastructural facilities were there in the villages, like related to the drinking water purification technology. In Maharashtra, the village, Saji village, which I had visited, they were focusing on purification of drinking water and providing safe drinking water to villagers. So they had one water ATM kind of facility in the village.
Wonderful.
In which they had a smart card, sir. Every household was issued a smart card. They were swiping it and getting means their daily usage of 5 liters or 10 liters of water. So it was safe drinking water, and means distribution was like justified for every household according to member. They had that issued smart cards. So it was very pronounced, and even in Maharashtra, Aadhaar card based ration distribution system was there. So certain things were very much like in systematic in Maharashtra.
Thank you very much. But when we talk of infrastructure, you mentioned infrastructure also. I'll just like to share that during pre-Green Revolution period, the extension specialists of PAU used to travel on bicycle or on buses and stay during night with the farmers. So the dedication and devotion of us has also gone down. Not that it is only infrastructure. They used to stay during night. Many people from Aishar must have known Dr. Takht Singh Sol, who went to NDA later on from PAU. Anyway, thank you very much. Very good presentation. Wonderful.
Thank you, sir. Can we have the question from Director, sir?
Well, Dr. Dahuja, no question. I just want to compliment all my faculty members and students. We are 782 at present online, and I want to see this touch 1,000, the maximum capacity. And the same enthusiasm is maintained tomorrow and day after tomorrow and in all the programs, please. So thank you, heartfelt thanks to each one of you. Thank you so much.
Yeah, Dr. A.K. Singh, I was also seeing this and I was feeling very happy. I was very happy yesterday, but today I'm over very happy. 788 or 89. But I have one suggestion or request. Students saying very good, very good, very good. Then I stopped seeing the chats. if I'm speaking, you will say very good. If you are speaking, I will say very good, because we are class fellows. So if such information doesn't come into chats, faculty is welcome. Then I will go on continue. Yesterday I continued. So if this can be,
there are comments in chat back, sir. They are coming, and people are appreciating presentations and also comments.
There are more. There is more appreciation.
Yeah.
And there are some very good comments. There is from Plant Breeder. Someone has asked that I have unmuted myself. I want to ask a question. But balle balle se kaam nahi chalta hai bhai. Chalo, anyway, we must appreciate also, but both things in a balance.
Thank you, sir. So we'll move on to the next presentation, which is by Mr. Posik Banerjee from the discipline of Agriculture Physics.
respected jury member and distinguished scientist, and dear friends, I welcome you all in my presentation. In the beginning, the Almighty created this vast universe. Then He created the beautiful planet, the Earth, where the plant grows and produces food to us. However, in its growing season, it faces scorching heat of sun and sometimes high rainfall. Thus, the life begins under the above stress conditions like temperature stress, moisture stress, salinity stress, and nutrient stress, and drought. This causes morphological, physiological, and biochemical changes within the plant system. The interaction of genotype with environment gives rise to phenotype, which ultimately affects the yield under the above stress conditions. And this phenotypic database is not so well developed. We all know that plant cannot speak, but it responds to the environment in which it is growing. And this is needed to be tested using imaging technology and collecting biophysical parameters. And this is important for their characterization and identification of genotype that can tolerate limited input supply under the face of climate change. Keeping all in background, I worked on the topic thermal imaging for characterization of abiotic stresses in wheat under field condition. The following objective: to identify, develop, or explore index using multivariate techniques for characterizing different abiotic stresses in wheat under field conditions, and to evaluate thermal and RGB image-based indices for discrimination of wheat genotypes and their relationship with the biophysical parameters under different abiotic stress conditions, and to explore the potentiality of RGB image-based indices in monitoring drought using satellite imagery in wheat growing regions. We experimented on 10 wheat genotypes which were grown in water stress, where deficit irrigation was applied on the basis of IW/CP ratio. Then we maintained a temperature of 3 degrees centigrade than ambient in a controlled environment for applying the temperature stress. And under nitrogen stress, we only applied 1/3 of the total nitrogen at basal. And for salinity stress, irrigation water of EC 4 deciliters per meter was applied. To understand the response of every genotypes, we took readings of biophysical parameters at different growth stages. Normalized dataset was used for developing new screening index using the principal component analysis method and side-by-side heat mapping and clustering. We scored the genotype based on their response, and thus their tolerance ability to abiotic stress conditions was assessed. Under water stress conditions, our newly developed screening index NWSTI identified genotype C306 as the most tolerant with the screening score more than 0.66 and PBW343 as the most sensitive with the screening value less than 0.33. Here, RWC, stomatal conductance, transpiration, and MSI perform significantly in screening them. Clustering and heat mapping also showed the similar result as found earlier. Simultaneously, NWSTI also gave high and significant R-square value with the crop yield and harvested biomass. Similarly, under temperature stress conditions, our newly developed index NTSTI picked out variety HD2985 as the most tolerant and HD2987 as the most sensitive under the temperature stress conditions. Here, photosynthesis, conductance, transpiration, and MSI perform significantly in screening those genotypes. Similar result was found in clustering and heat mapping the genotypes. Additionally, NTSTI also showed high and significant R-square with the crop yield and harvested biomass. Under nitrogen stress conditions, our screening index NNSTI identified HD2967 as the most tolerant and HD3043 as the most sensitive with the corresponding score values. Here, NDVI, conductance, transpiration, and MSI perform significantly in screening them. And the result was found at par after the clustering and heat mapping. Additionally, NNSTI also showed high and significant R-square value with the yield and crop biomass. We screened 49 wheat genotypes under the salinity stress conditions. Here we found ASN17 as the most sensitive and KRL302/340 as the most tolerant. And here photosynthesis, stomatal conductance, transpiration, and leaf sodium content perform significantly. A similar result was found after heat mapping and clustering, and here also our index NSSTI showed significant and high R-square with the yield and biomass. Now, why these indices are performing well in screening genotypes? Here, responses of each genotypes coming due to interaction with the environment are given certain weightages, and we combine them all to a single factor which helps in screening them. And this concept was accepted for publication in Ecological Indicators. Thus, newly proposed indices like NWSTI, NTSTI, NNSTI, and NSSTI performing satisfactorily in screening wheat genotypes, and they also showed significant relationship with the yield and biomass. Biophysical parameters identified through PCA will be useful in improving the biotic stress tolerance ability through breeding strategy. Is it possible to discriminate a large number of wheat genotypes under field conditions using the imaging technology, which is fast and non-destructive? We experimented on this in this objective. For this, we took the thermal and RGB images under field conditions of wheat genotypes and extracted thermal and RGB image data and marks the soil part for calculating the vegetation indices. Thermal and RGB images of wheat crop are shown here at clearing and flowering stage under water stress condition. And to understand the crop condition, we calculated 14 different image-based indices. Performance of the image-based indices are also observed in different stages under water stress condition. Sensitive image-based indices were identified by correlation coefficient analysis with the NWSTI. Here we found CWSI and IG in thermal and GRVI, excess gain, excess read, etc. in RGB significant. We took the ratio value of RGB and canopy temperature for discriminating the wheat genotypes. Performance of sensitive thermal and RGB image-based indices under water stress conditions are shown here, which discriminated wheat genotypes based on the water stress tolerance ability. Here, all the image-based indices showed significant relationship with the stomatal conductance and transpiration rate. Similarly, for the temperature stress, thermal and RGB images were taken. And here, sensitive indices, as shown here, discriminated wheat genotypes into 3 groups. In general, among different biological parameters, stomatal conductance and transpiration and RWC showed significant relationship with the image-based indices. For nitrogen stress, images were taken at different growth stages. And sensitive indices discriminated genotypes into 3 clusters. Here, NDBI, photosynthesis, transpiration, MSI showed significant relationship with the image-based indices. Under salinity stress, we took images of 49 wheat genotypes at different stages. Sensitive indices discriminated them into 3 clusters. Distinct location of tolerant and sensitive genotypic groups showed the ability of the image-based indices in their discrimination. Thus, combination of thermal and RGB images is able to discriminate wheat genotypes under different abatic stress conditions, because under stress condition canopy temperature will increase and values of vegetation indices will decrease. Thus, the ratio of vegetation indices versus canopy temperature will be low for the sensitive and high for the tolerant, and this helps in their discrimination. We found variation in responses in RGB and thermal image-based indices under different stress conditions. Mathematically combining thermal and RGB image information found suitable in discriminating the wheat genotypes, and vegetation indices that perform significantly in genotypic discrimination could be useful in monitoring abatic stress from satellite imagery covering a larger geographical area. Under third objective, we explored the potentiality of RGB image-based indices in monitoring drought using satellite imagery in wheat-growing regions. Image-based vegetation indices, which are found sensitive in genotypic discrimination under water stress conditions at the ground level, are upscaled here for monitoring drought in 3 wheat-growing regions of Rajasthan. We took time series image data and rainfall data and used them in drought analysis and yield estimation. MODIS Terra imagery data in red, green, blue, and NR bands were collected at 8 days interval for the last 20 years, and district-wise wheat area masking was done. Then we observe the variation in different vegetation indices like GRVI, VEG, excess red, excess GR, excess green, COM, NDVI, and VCI for the 3 wheat-growing districts, that is Ganganagar, Hanumangarh, and Alwar, and simultaneously took rainfall data for the last 70 years for calculating SPI and identifying drought year for those 3 districts. SPI was calculated for the month of February and March, which coincide pre-flowering, flowering, and post-flowering stages of wheat. Based on the SPI values, we identified extreme, severe, and moderate drought years both for the month of February and March for the 3 districts. Similarly, train adjustment was done for monitoring drought in different years. In order to separate the effect of weather from technology, a modification, that is the train adjustment, was done. For example, year 2009 was drought year, but VCI could not pick it as shown by the value of 0.4. In contrast, after the train adjustment, VCI clearly identifies the year 2009 as drought, as shown by the value of less than 0.35. Variations in trend adjusted vegetation indices were observed, and drought years were identified as shown here. These trend adjusted vegetation indices also showed significant relationship with the SPI4 both for the month of February and March for the Alwar district. Thus, the drought year identified through the vegetation indices are showing similar pattern as it was identified through SPI, which shows the potentiality of these indices in monitoring drought. To use the vegetation indices in yield estimation, we regressed them with the wheat yield and found significant R-square value for each of them. Then we predicted the wheat yield using 10 different models as shown here. We did calibration and validation each of the model with the available dataset. Comparison among the model was done using different statistical parameters as shown in this table. In the calibration stage, QRF was found the best, but in the validation stage, SVM was found the best for yield estimation with the lower RMC value. Thus, we found significant relationship between the SPI4 and other vegetation indices for monitoring drought over the wheat growing regions. Here, Alwar showed the best result for monitoring the drought for the month of February and March. And among the 10 multivariate models, SVM was found the best in predicting wheat yield using the vegetation indices. Thus, we developed new screening indices and tested them in identifying tolerant, moderate, and sensitive wheat genotypes grown under different abative stress conditions. We identified sensitive thermal and RGB image-based vegetation indices and combined them for discriminating wheat genotypes grown under different abative stress conditions through rapid and non-destructive way. We upscaled the vegetation indices from field level to satellite for monitoring drought in wheat growing region in Rajasthan and estimated the wheat yield using the vegetation indices where SVM was found the best. The methodology developed would help in high throughput phenomics of different crops in other regions too. Indices developed here can be used for screening large number of genotypes for the other crops. Sensitive indices can be used in agriculture for abative stress detection and precision farming. Vegetation indices examined on the satellite level can be used for devising drought monitoring criteria. Due to the journal of ecological indicators with an ascending of 10.96 and developed two processes. I have participated in different symposium and conference and represented myself as BOS in my division. I sincerely acknowledge my chairperson and advisory committee, all the supporting member, Division of Agricultural Physics, PG School IRI for providing the financial assistance and my parents and friends. And I thank you, all of you, for your kind patience.
We'd first like to hear from Dr. Bansal, sir.
Okay, thank you. And Mr. Your name is Mr.
Kaushik.
Mr. Posik Banerjee.
Banerjee.
Yeah, Mr. Posik Banerjee. Yeah, excellent presentation and very good piece of work.
Thank you.
Think, which is certainly required. It has also added value, to the existing technology. I believe so. Maybe I will have some clarifications from you. You started with talking about developing these, particular indices, based on some 10 genotypes of wheat to start with.
Yes, sir.
While you don't know anything about what you're going to develop, right? Whether it's going to differentiate sensitive versus tolerant genotypes or not, we have no idea, right? I mean, when you started. So was it not important that you have developed, this indices, for example, for whether you talk of nutrient stress or water or temperature or even salinity for that matter, with a single genotype, with well established kind of a stress under field conditions. I'm happy that you did all your experiments in the field conditions, which is more value, no doubt about it. So my question is, that, and then later, whatever you got the observations, at different stages and all of development in one genotype, then you would have tested it. Your methodology, of all different kinds of air, kind of monitoring, prediction, and etc. in large number of genotypes where you already know, from the experience of breeders and otherwise, that these are, for example, you discovered C206 is tolerant and, PW343 sensitive. That should have come later, which you already know that it is there, but then you could have taken a large number of genotypes. So what is your comment on this before I go to the next and few more questions?
Yes, sir. Thank you, sir, for your question. So actually, this study has some pre-experiment where we develop only single genotypes under 4 release conditions, which was done before. And we tested all these things here also. And after that, we applied the same methodology for discriminating all these genotypes as I have shown in this case. And before that, we also searched different literature what is the genetic potentiality of all the genotypes, but not the all. And we got some kind of that information. Yes, this kind of genotypes are some sensitive to tolerant or medium range water stress tolerance or temperature tolerance potentiality. So I got some idea from them also. And vis-a-vis, I also conducted my own experiment under different irrigation treatment. And firstly, we identified those things and developed those indices and applied then thereafter for the water condition.
Okay, did you also measure, Mr. Kaushik, the level of stress? Level of stress, did you quantify it? Whether you're talking of nutrient stress or drought or high temperature or salinity for that, did you measure that under what level stress are you working?
Yes, sir. For measurement of the stress means we actually depend on the crop performances, how they are performing under that stress.
No, performance is fine. But did you quantify that what was the level of stress the plants were exposed? Though you were not able to control, I can understand probably because they were under field condition. But did you still quantify that? And the question would be related to this, that up to what level of stress your indices could work? How far can you stretch the kind of intensity or duration of stress of a given type?
Okay, sir. In this case, for the water stress, I have applied only irrigation at CLS stress. Thereafter, I have not applied anything. And based on that irrigation level, they performed actually. All the other.
No, I agree. You imposed stress, but you didn't measure the soil percentage volume or in the leaf or so that relate. That your methodology is good for this particular level of stress. Anyway, that's one. Second, already, of course, we know through the excellent work of, of course, in our own division at IRI, Dr. Vishnathan and others, we have got high throughput phenotyping facility already created. But I'm happy again that you did all under field experimentation, which is more important. But even then, scientifically to prove your hypothesis right or to be having more robust confidence on it, did you make a comparative analysis? With the available data on some of those genotypes that you used, and how strong confidence do you have in the kind of indices you have developed to be utilized better than others?
Yes, sir. I have compared it with the published literature, which also work in the same line under field condition also. And we compare our result with those literatures actually for different varieties like variety C306, PW343, and HD295. They also show accurate result as we found in our study, sir. But they also develop these indices work, and their indices are also to some extent matching with our results, sir.
Okay, we have to move on. Thank you. Thank you, Mr. Kaushik. All the best to you.
Thank you.
Thank you, sir. Now I would like to take up the question from Dr. Ravinder Kaur, ma'am, because she raised hand first.
Thank you, Dahuja-ji. Mr. Kaushik, excellent presentation. At the outset, I think I must congratulate you for doing 3-in-1 PhDs. I could see 3 to 4 PhD titles running in one PhD. You must have really worked hard in these days. You started with developing remote sensing indicators for discriminating between different wheat genotypes with respect to their sensitivity to stress, water and salt both. Then you said that you upscaled it and used it for drought monitoring. Then you said you also used it for wheat prediction, wheat crop yield prediction, right? So these are like 3 dissertations in one. And I feel that the reason why you are not able to answer precisely is because of that only. My question is that I could not see that of the various 13 indices which you validated for having remarkable potential with respect to stress, which were the ones which you upscaled for drought monitoring? I could see only VCI. And that's a conventional indicator which we use. So what was the use of objective number 1? Precisely, that's the question.
So, thank you, ma'am. In our objective 1, we only use the thermal band and the RGB band, where we calculate different indices and found sensitive for genotypic discrimination in that objective. And when we are upscaling those indices at the satellite level,
Then we also took the NR band also, as well as our sensitive band. Which
I could see in drought monitoring only VCI validation. You use VCI SPI for drought.
VCI
isn't
as
it?
well as,
So
yes,
where is
sir.
the thermal in VCI?
No, sir. Ma'am, actually, in the satellite, I have not used the thermal band. Only RGB
So
NR
that is the question, Kaushik, we'll stop here. My message is that your 3 objectives of one dissertation, these 3 objectives should lead you to a goal. What was your goal? If the objective 1 is not flowing into objective 2 and 2 not flowing into 3 to make you reach the goal, why were there 3 objectives? They were like 3 theses.
No.
Okay, so it's just my comment, not question. Thank you very much.
Thank you, ma'am. So we'd like to hear from Dr. Patra, please.
Thank you, Dr. Posik Banerjee. Again, excellent work you have done, and lot of information you have generated. Some of these slides, it was very difficult, in a very short time to go through and to grasp what exactly you are presenting. So that is, I'm not criticizing, but because a lot of information you have generated, and excellent presentation, lot of informations. You have addressed 4 very important stresses: high temperature, water, nutrient, and salinity. Just to know what were the degrees of stresses. For example, the nutrient stresses, how much it was. Since Professor Bansal Sahab also asked about, perhaps you have not seen really in the soil, what was the level of the nutrients, particularly you worked on the nitrogen, what kind of stresses, what was the availability, how it was changing, and how it was impacting to the yields. So that is also still, I'm trying to understand. And another thing, all the abiotic stresses you have measured, are these indices able to distinguish the different stresses, when they are occurring sometimes at a time? And another is for the different stresses, can the screening values, are there changes, screening values for the different stresses? So these are the few queries from you, Dr. Posik.
Thank you, sir. Sir, I have observed the changes in the soil water content at depth wise for defined experimental plot I have conducted at our field. And sir, regarding your second questions, that whether these indices are able to
water, not the nutrient also I asked you.
And for the nitrogen stresses, we have applied only the basal dose of nitrogen. And here also we monitor the level of nitrogen stress in terms of biological parameters, sir, how they are responding in terms of the stresses. And sir, whether these indices are able to discriminate temperature stress from the water stress, for this we need to have some separate experiment. Actually, if we have all the experiment in one plot, then it will be able to do that kind of investigation very easily. So for this we need some separate experimentation, and data is also needed to generate in that way. Actually.
Okay, since the time is short, we can discuss later on when we will meet. All the best. All the best. Thank you.
Thank you, sir. Can we have the question from director, sir?
Thank you, Kaushik, for excellent presentation, and very well you have answered the question. I have just one remark, and this should not be, in any way coming into the decision making. The first 2 slides that you presented, exactly with the same language they were presented yesterday by an MSc student. If I remember correctly, exactly the same 2 slides were presented. He also talked about environment. He also talked about, the global population increase and all those things. It was exactly the duplicate slide. So I think these things should be avoided. I don't know whether who has done it, whether the original thing is with you or with the other student, but this should be avoided. And I remember word by word, it was exactly similar.
Thank you, sir, for your observation.
Okay.
May we have the comments from the chairperson, sir?
Yeah, I will also, again in lighter vein, again, Almighty created. Do we believe in evolution or not? I believe in God and I also believe in evolution. So it is for Dr. Banerjee. First of all, Dr. Banerjee, very good work. Wonderful presentation.
Thank you,
A
sir.
new field which is emerging, which is important because we have to screen or evaluate germplasm on as large scale as possible. But again, so first sentence you said was Almighty created, and then you said plant don't say wheat plant breeder and maybe Dr. A.K. Singh also. Yeah, what Dr. Norman Borlaug said. Plant do speak if you listen to them. So avoid such things.
But in whispers, and they can only those can hear who remain in close contact with them. Not everybody.
So,
what is it? Sir, permit me to leave for a time. I have to join that CG meeting. The RISE presentation is coming, so I have been asked to be there during RISE presentation. So I beg your pardon. I will join back after that sometime.
Okay. That is also important, please. So then another comment was about slides. The normal principle is which I 6 by 6 columns, 6 rows. But too much. Just imagine that. Can you read the slide when you are presenting? If not, then how you expect someone, the third person, who is seeing the slide? Then many slides were tilted. If you have done that, if you have that artistic mind. Excuse me?
No, sir, I have muted him.
Now let us listen.
No, I think it was by mistake.
Okay. So many slides are tilted. If Dr. Banerjee, if you are of artistic mind, it's very good. But if it is just you did. So now coming to the question, what was the basis of selection of genotypes?
Thank you, sir. Sir, we studied the genetic potentiality and other natures of those genotypes published in annual report of different institute, and we found various responses of those genotypes, not all, but some of them, and we choose them and experimented them in our plots, sir. And they are also having good yield potentiality, covering Indo-Gangetic plain and northwestern plain of India, and that's why.
Okay. Let me move on further. We know whenever we talk of any photographic canopy temperature, etc., height and leafiness are two very important characters. That way, C306 falls in another category. And now, of course, data analysis techniques have come. But normally, we need normal distribution of data. And once you include C306 an outlier. And most of the good universities I have not seen in India, but outside they do first outlier test before going for any analysis. So C06, when you have that will definitely. So selection of genotype is very, very important. Equally important is the level of stress, what Dr. Bansal sir mentioned in the beginning. Because if there is linear relationship, fine. But mostly linear relationship is not there. Then coming to Northwest India, it is, I will say, for future work. Flooding has become important in wheat, flood tolerance. This year we have extreme rains. Now, at least in Punjab, Haryana, Western UP. It is, and I'm very happy that you did work under field condition. So my final and most important question is, I don't know who gentleman is in charge, name of the gentleman who is in charge at IR in wheat program. But the earlier gentleman, Dr. G.P. Singh, who's at Karnal for last some years, when he will adopt your procedure, your technique, your indices, when do you expect he will adopt?
It's Dr. Rajveer Yadav.
Dr. Yadav. So when Dr. Yadav will adopt these techniques? So I modify my question. For first Dr. Rajveer, then Dr. G.P. Singh, then PAU and HAU and Pantanagar, all equidistance. Merit also. What are your expectations?
Yes, sir. These indices that we have followed the methodology is common, and it is developed on pilot basis, and it can be used for any other things. It means we can fit other genotypes and other,
When you will convince Dr. Rajveer Singh Yadav to use it, when you will convince him to use it.
Yes, sir. Actually, sir, these indices, the screening indices, actually, it is applicable to all. Means the methodology developed is common one actually. And parameters.
Question is how quickly can we start doing it? That's what the chairman is asking. Can we use it tomorrow?
Oh no, so actually for that we need to wait for taking the observations first. And if you use the imaging technology, then it can be done, sir, actually.
I ask you a hard question. Maybe faculty colleague can respond. The level of confidence, your publication, and apply something in the field. Anyway, very good, wonderful. Dr. Dhuja, we can move forward.
Thank you, sir. So next presentation is by Ms. Sonaka Ghosh from Discipline of Agronomy.
According to Professor M.S. Swaminathan, if conservation of natural resources goes wrong, nothing else will go right. There are many challenges of the existing agricultural practices. The sustainability of intensively cultivated rice wheat cropping system of the Indo-Gangetic plains has been reported at stake due to slowdown in yield growth rate, which is associated with many challenges. Indian soils are currently witnessing nutrient imbalances. The map shows wide variation in fertilizer nitrogen use in different states of India. 54% of India's groundwater wells are decreasing. The reports on looming water crisis warn for improved water management. Residue burning is a serious concern causing environmental pollution. The paper highlights that the farmers can get more profit by switching from residue burning to use of happy seeder. About 27% of global greenhouse gas emissions occur from agriculture sector. Weeds have emerged as a global agricultural pest. Weeds cause economic loss of about $11 billion in 10 major crops of India. To address these challenges, what is the solution? The solution could be conservation agriculture. It is being promoted and adopted for sustainable crop intensification. It comprises the practical application of 3 interlinked principles such as no or minimum mechanical soil disturbance, biomass mulch soil cover, and crop species diversification in conjunction with other complementary good agricultural practices. Conservation agriculture has been reported to meet 5 sustainable development goals. Due to such potential benefits, it is being practiced globally on about 205 million hectares of cropland. There are some researchable issues under conservation agriculture. One major issue is optimization of nitrogen fertilization in conservation agriculture. The picture shows the driving link between nitrogen and the sustainable development goals through optimum nitrogen use. The other major issue is weed dynamics under conservation agriculture. Weeds are the major challenges under conservation agriculture. Therefore, to achieve sustainable crop production with reduced negative effects on soil and atmosphere, I have conducted an experiment on nitrogen management and weed dynamics in a conservation agriculture based maize wheat mung bean system during 2018-19 and 19-20. The objectives of my research were comprised of to appraise the conservation agriculture effects on weed seed bank, to assess CA effects on crop productivity and profitability, to work out the use efficiency of water, nutrient and energy across the treatments, and to assess CA effects on selected soil physical, chemical and biological parameters and for mitigation of greenhouse gases. The present experiment was conducted in the 9th and 10th year of a long-term conservation agriculture experiment. It was conducted in a randomized complete block design with 10 treatments and 3 replications. In this system, different CA-based practices were compared with conventional tillage practice. Different methods of crop establishment such as permanent narrow bed, broad bed and flat bed with and without residue retention of maize, wheat and mung bean along with 75% and 100% recommended doses of nitrogen were integrated in CA practices. A total of 15 weed species emerged in the soil seed bank, 8 of which were broad-leaved weeds, 5 grassy weeds and 2 sedges. The CA-based practices saw dominance of broad-leaved weeds. The rest of the weeds were found higher under conventional tillage practice. The weed seed bank density was estimated by seedling emergence method on pot culture study at 2 depths of soil, that is 0 to 7.5 cm and 7.5 to 15 cm. The total weed seedling emergence was higher under conventional tillage practice. Permanent narrow bed and permanent broad bed with residue with 100% nitrogen significantly reduced the weed seed density. Crop residue retention resulted in 25 to 48% reduction in soil weed seed bank at 0 to 15 cm soil layer compared to conventional tillage practice. Maximum reduction of weed seed density was observed under permanent narrow bed and permanent broad bed with residue with 100% nitrogen. To know the weed dynamics in field condition, category-wise weed density and biomass was estimated. The field study indicated the dominance of broad-leaved weeds under CA-based practices in maize, wheat and mung bean. The dry matter accumulation and root length density of the crops were negatively correlated with increase in total weed biomass. The CA-based residue return practices improved grain yield of crops due to weed suppressive effects of residue retention, its long-term favorable effects and better crop growth. These practices resulted in 16 to 22% higher maize grain yield, 19 to 27% higher wheat yield, and 23 to 35% higher mung bean yield compared to conventional tillage. Among these practices, the treatment permanent broad bed with residue with 100% nitrogen was found superior and remained comparable with other residue return practices. The system productivity was evaluated in terms of wheat grain equivalent yield. The CA-based practices resulted in 24 to 31% higher system productivity compared to conventional tillage. The treatments permanent broad bed and flat bed with residue with 100% nitrogen were found superior. The increased yield under CA-based practices resulted in 35 to 47% higher net returns compared to conventional tillage. The treatment permanent broad bed with residue with 100% and 75% nitrogen remained comparable in registering higher system productivity as well as net returns. Permanent broad bed with residue resulted in about 25% savings in irrigation water use compared to conventional tillage. The treatment permanent broad bed with residue with 100% nitrogen registered 61% higher water productivity than conventional tillage and remained comparable with permanent broad bed with residue with 75% nitrogen. Improved crop establishment practice combined with decreased irrigation water use resulted in improved water productivity under broad bed practice. Permanent broad bed with residue with 100% nitrogen led to about 31% higher partial factor productivity of nitrogen compared to conventional tillage. However, significantly higher partial factor productivity of nitrogen was registered under permanent broad bed and flat bed with residue with 75% nitrogen due to lower rate of nitrogen application and comparable yield gains with 100% nitrogen. The CA-based residue return practices resulted in 17 to 23% higher output energy than conventional tillage. Among them, permanent broad bed and flat bed with residue with 100% nitrogen were found superior. But the CA-based residue removal practices obtained significantly higher net energy return due to energy savings in terms of no residue application. But crop residue, being renewable energy resource, has various advantages when returned to soil in terms of increased crop yield and enhanced soil health. Permanent broad bed with residue with 100% nitrogen registered about 38% higher soil mineral nitrogen than conventional tillage and found comparable with other residue return practices. It was observed that the CA-based residue return practices had higher total biomass content, including both above-ground crop residues and respective root biomass, which resulted in maximum carbon input in these practices. It led to improvement in total carbon percentage in these practices. Compared to conventional tillage, the residue return practices increased 40 to 54% of total carbon percentage at 0 to 5 cm, 40 to 46% at 5 to 15 cm, and 30 to 38% at 15 to 30 cm soil depth. Among them, permanent broad bed with residue and flat bed with residue, irrespective of varied nitrogen application, were found superior. Improved aggregation in CA-based systems increased the physical protection of soil organic carbon in soil aggregates. It was observed that the phospholipid fatty acid mediated microbial biomass positively correlated with the increase in total carbon percentage in the soil, which indicated the positive influence of crop residue retention on soil microbial functions. The soil aggregate stability in this study followed the similar trend to total carbon content in soil, as soil organic carbon is an important factor in the development of water-stable aggregates as a soil particle binding agent. Permanent broadbed with residue with 100% nitrogen significantly registered higher water-stable aggregate percentage in 0 to 5, 5 to 15 and 15 to 30 cm depths of soil and remained comparable with other residue return practices. The CA-based residue return practices resulted in significant reductions in carbon dioxide emissions. The absence of tillage activities, improved soil aggregation, and the protection of soil organic carbon in soil aggregates under conservation agriculture might have resulted in less carbon dioxide emission. However, highest nitrous oxide emission was observed under flat bed with residue compared to conventional tillage and other bed planting practices. The lowest global warming potential was recorded under permanent narrow bed and permanent broad bed with residue. The global warming potential was reduced to about 16% under permanent broad bed with residue compared to conventional tillage. From this study, it was concluded that permanent broadbed with residue with 100% nitrogen significantly reduced wood interference and improved crop productivity, profitability and resource use efficiency compared to conventional tillage and led to significant improvement in soil properties and reduced greenhouse gases. Application of 100% and 75% nitrogen with this treatment was found comparable, indicating a saving of 75 kg nitrogen per hectare in the year-round cropping of maize, wheat and mung bean. Hence, the CA-based permanent broadbed with residue with 100% nitrogen in the initial years of adoption and 75% nitrogen over a long period may be recommended for sustainable intensification of maize wheat mung bean system in the Indo-Gangetic plains of India and similar agroecologies of the tropics and subtropics. The future line of work is to conduct periodical weed dynamics over a long period, residue quantification and decomposition rate constant determination, soil nutrient stratification and moisture dynamics, and GHG footprint. These are my publications. I have published 4 research articles, 5 review articles, and 1 popular article. We have developed a herbicide calculator web application. For this research, I received DST Inspire Fellowship. I also received Student Travel Grant Award by Indian Society of Weed Science Golden Jubilee International Conference at ICR DWR Jabalpur. I have participated in oral and poster presentations in many international and national conferences. I am also an active member in Indian Society of Weed Science and Indian Society of Agronomy. Finally, I would like to express my sincere thanks and gratitude to my Chairman and Advisory Committee, Head, Professor and Faculty Division of Agronomy, Director, Dean and Joint Director Education, PG School ICRIRA New Delhi and DST Inspire Fellowship. Thank you everyone.
Can we have the comments from Dr. Bansal first?
Yeah, okay, thank you. Your name is Ms. Naka, right?
Yes, sir. Good morning, sir.
I think it's an excellent piece of work. And certainly which is important, for saving our natural resources. At the same time, saving our, energy and, even it becomes profitable to the farmers in the long run. I'm happy that it was a kind of a comprehensive study. You measure all the other important parameters. But a few questions to start with, for clarification. Did you do the work in 1 season or 2 seasons or 3 seasons?
Sir, it was conducted in 2 years, sir, in 3 crop seasons, maize, wheat, and mung bean, sir.
3 crop?
Yes,
No,
sir.
I mean, continuously for 3 crop seasons.
Yes, sir, continuously for 3 crop season in consecutive 2 years.
Okay, and what was your plot size? Because you're comparing data, estimating data all per hectare basis. So what was your size of the plot where you carried all these?
Sir, the length of the plot was 30-meter length and 8.4-meter width, sir.
So was it enough, you think, for such an analysis you have done later, your conversion or per hectare basis?
Yes, sir. We have.
You for example, you're talking of water productivity. You calculated kg per hectare.
Yes, sir.
Put energy, mj per hectare. Even greenhouse gas emissions, which is good that you measured all this, which is important. But you again kg per hectare. How did you measure greenhouse gas emissions?
Sir, by closed gas chamber technique, it was collected and measured through gas chromatography.
Now that's a standard methodology you must have used.
Yes, sir.
Understand. But the question is again, you talked about mostly nitrogen and weed management.
Yes, sir.
So why didn't you include water as well?
Sir,
these crops are mostly, of course, grown under irrigated conditions. But even then, or did you estimate any savings in water as a result of your conservation practices, which you found the best?
Yes, sir. The permanent bed planting practices showed about 25% savings in irrigation water use compared to conventional tillage practice, sir.
And another question is, see, you've taken, of course, the standard variety of any of these 3 crops.
Yes, sir.
Discussion with are they ones which are already high yielding kind of genotypes, or would you not like to also see the role of, you're an agronomist, fine, but you still not like to see the role the seed would play, an improved variety, for example, in conservation agriculture, that it could even do better under less nitrogen, with less water, or will have better ability to emerge faster, have less competition, compared to with weeds growth, etc. So what is your message about this, that how a seed can also play a role, or improved seed variety, I would say, can play a role?
Yes, sir. Improved seed variety along with better management practices under conservation agriculture can play a potential role for effective weed suppression as well as for increased yield, sir.
No, but has it found any place in your study? Why it didn't find a place? I mean, you started, of course, I can understand, there must be some limit to your experimentation, but still.
Sir,
yeah.
Yes, sir. On a single variety means not I have not studied in different varieties of crops. A fixed variety of crop, I have seen the effects, sir.
Okay, and did you observe or any kind of a effect which would have nullified as a result of some other abiotic and biotic stresses during your cropping season? Did you, by the way, quantify that to nullify the effect of those, biotic or abiotic stress factors?
Sir, weed seed bank reduction can be done by long-term conservation, adoption of conservation agricultural practices, because weed seeds mostly remain in the top layer of the soils under conservation agriculture practice. So by predation and weather fluctuation and increased weed emergence over the years can deplete the weed seed bank. And also, sir, by proper residue quantification, the pest incidence also can be minimized compared to other practices.
Okay, thank you.
Thank you, sir.
Thank you, sir. So let's hear from Dr. Patra, please. Sir, you're on mute.
Yes. Thank you very much, Dr. Sunaka Ghosh.
Yes, sir.
Excellent presentation, and we know about the conservation agriculture at IRI. It is an excellent piece of research going on for a long time now. It is 10 years, and a lot of publications have come out from this study, and I frequently visit the site, so I know. So congratulations to you and the team, the chairman, for continuing so excellently this site. And most of the things, we discussed many times, but since you presented and for the PhD, which you made so well, just, maybe, it's in agriculture, how much area of conservation agriculture in India. So this is one. Another is the weed dynamics. Your focus was on nitrogen and the weed dynamics, because you cannot take so many things, what are this and that, so that is not possible in a short time, and that too also in COVID situation. Last 2 years we are facing. So weed dynamics, what was before, in the initial years, first year, second year, third years, maybe those things have been recorded by the PI of the experiment, and now what you see after 10 years, what was the dynamics, may not be at the different depths, but maybe at least what was there at that time identified, and then cataloging those weeds, and now so we can see, understand the dynamic, anything, some problematic weeds came, which is difficult to control, manage, and the pest and diseases, the biotic stresses, Professor Bansal have already asked you, because there is also things which is seen after some time, the pest and diseases, because of the residues and other things spread on the soil. So, and about the greenhouse gases, I hope you are noting my queries.
Yes,
The greenhouse gases, very good, and we all know, but I like to know from you why the greenhouse gases emissions are low in conservation. The explanations from you, I want to know. And then the carbon, which is extremely important for carbon sequestration, and conservation agriculture is very much potential solutions for that, for more carbon sequestration. In this, many of the studies reporting that the beyond the 10 centimeter depth, the carbon accumulation is very, very low, but in your case, it is more. So you must have compared that many of the studies, even in Nature also publication came that it is confined in the 0 to 5 centimeter or so depth, but you got more in the lower depth also, 10 to 15, 15 to beyond 15 centimeter. What may be the reasons? So these are not very difficult questions, and you must have studied, but I'd like to hear from you.
Sir, regarding your first question about adoption of conservation agriculture practices in India, so it is about 1.5. So it is about 1.5 million hectares in India. And sir, regarding your second question about weed dynamics in the field, sir, in the initial years of adoption, weeds are the major problem, and in this field, sir, perennial weeds are a major problem in the initial years of adoption. But now I have observed in my field, the weed dynamics has been shifted to annual broad-leaved weeds. So perennial weeds have been reduced, and the problem has now come to even grassy weeds also have been reduced compared to annual broad-leaved weeds. And if we say consider the total weed seed bank in the seed bank as well as in the field study, it was comparatively less and compared to conventional tillage practice in residue return practices. And regarding your third question, regarding sir, greenhouse gases emissions, sir, these are significantly lower carbon dioxide emission under conservation agriculture practices is due to absence of tillage activities, crop residue retention, and the protection of soil organic carbon. And I also told about higher nitrous oxide emission in flatbed with residue compared to residue return bed planting practices and conventional tillage. It is because of higher substrate availability and moisture content in flatbed compared to bed planting practices, due to as bed planting practices require less water compared to flat bed planting. So in that scenario, the nitrous oxide emission is higher in case of flatbed residue. So if we reduce maintain or optimize the water requirement as well as quantify the proper amount of residue, we can reduce the nitrous oxide emission also in conservation agriculture. And sir, regarding your fourth question of increase in total organic carbon in the soils, compared to surface layer, the total carbon is decreasing in the lower depths of soil, but it is found higher in that of conventional tillage, because it is a long-term conservation agriculture practice, and I have conducted in the 9th and 10th year of experiment. And over the years, the residue, the 3 crops residues, including the legume crop and also the respective root biomass, might have accumulated sufficient organic matter to have a higher organic carbon in these practices.
Very nice, excellent. All the best.
Thank you, sir.
Thank you.
Thank you, sir. Now we'd like to have the question from Dr. Mansingh, sir.
और ऊपर शुरुआत में भी है.
Thank you. Sonaka, appreciate your presentation, but I take the
Thank you, sir.
3 points raised by first the chairperson, Dr. Hillo. He said that the PhD students knows the maximum. Dr. Patra's remark and Dr. K.C. Bansal's observation and my own observation in the IRI. What is the new thing which you have added in your PhD thesis, which is beyond several publications of T.K. Das and group? I know that on your advisory panel, one of our colleague from water, Dr. Sushma, is also adding to that. So I want to know that precise difference in this PhD program, which you do. Very specific. Yeah.
Yes, sir. Sir, I have studied the weed dynamics in both the soil seedbed as well as in the field study, which was very least studied in the conservation agriculture practices, like to quantify the amount of reduction and as well as the species-wise diversity, the weed dynamics actually, like what is the weed composition, etc. And also the greenhouse gas emissions under different practices, and quantifying water, energy, and nutrient. And also these are different improved physical, chemical, and biological parameters.
It was not synthesized in your conclusion slide. Thank you.
Okay, sir. Thank you, sir.
May we have the comments from Dr. Patil, please?
Very good presentation, Sonaka.
Thank you, sir.
Just you know that conservation agriculture improves the soil health, no doubt about that. My first just question to you, it is a highly technology-driven technology or less technology-driven technology? And secondly, in this conservation agriculture, sowing of seeds in an untilled land without removing of crop residue are in sharp contrast to the traditional beliefs of the farmers. So then how to convince the farmers to go for the conservation agriculture? Just highlight.
Yes, sir. The conservation agriculture practice is highly management intensive practice and machine intensive practice. So if the farmers should be given better subsidy, like we have sown the seeds in conservation agriculture plots by the help of turbo seeder, happy turbo seeder. So if the farmers have been given sufficient training and demonstration on the use of this machinery, and they if they will be given in the machinery at the subsidized rates, and also more number of custom hiring centers will be there, then I think they can better adopt the practice, sir.
Okay.
Thank you, sir.
Thank you, sir.
My turn now.
Yes.
So congratulations, Dr. Oh, Dr. Please. Dr. Kesing?
Dr. Kesing.
Please. Sorry.
Just a small submission. Dr. Sonaka,
yes, sir.
Division of Agricultural Economics, IARI, has done a lot of work on RCT, impact of RCT and economic and environmental impact they have studied, which includes Dr. Suresh Pal and others. Have you gone through those studies?
Sorry, sir, I have not gone through this study.
They have done on a large sample from the farmers' field, and you will find that there is significant difference in the yield enhancement, what you are reporting and what they are reporting. When you will review, you will find that they have reported maximum increase up to 10%. where I think in your case, even the income increment was, net return was increased by more than 46% and so on. So my only suggestion is that when IARI has done this kind of work, a lot of work, different RCTs, not only one technology, they have done this. So you should go through it and
yes, sir, I want to say something about it, sir. Actually, sir, many farmers are using partial conservation agriculture technology. Means like at least in one crop, they are using zero tillage technology, but the potential benefits of conservation agriculture can be realized by adopting the three principles: by crop residue retention, minimum soil disturbance, and along with retaining one legume crop in that system. And as it is a management intensive practice, better management practice can yield the higher potential in this system, sir. So so the farmers should be trained properly about its proper management and practices, better practices.
Okay, thank you, sir. So may we have the now comments from the chairperson, sir?
Thank you, Doctor. Sorry I interfered earlier. Next time I will wait for okay from you. So first general comment, what I said yesterday about growth rate. Please, we are dealing with biological system. As we move up, growth is bound to decline because there is potential limit in biological system. Moreover, growth rate is expressed as mean of the base of the as a percentage of the sum or regression coefficient and then percentage of the mean performance. So please, if you must want to say growth rate has declined, please say that as we are moving up. Let us not give, we give ourselves handle to the these bureaucrats. For 12 years, I taught them, and I'm happy that I have been successful in Punjab, telling them that there is no upper limit in wheat and rice where we can go. We have gone up to more than 50 quintal in wheat, average state average, and 69 quintal of paddy. What else PAU can do? So please don't say all the time growth rate has declined. Say that as we are moving up, growth is expected to decline, it has declined. So that is a first comment. Second is, Dr. Ghosh, your work is very good, wonderful.
Thank you, sir.
I will present your data in two ways. Comparison as you did and comparison in comparison to what was the original status, as earlier, I think Dr. Patel or Dr. Patra mentioned that what was the status in the beginning.
Yes, sir.
And increase over, yes, I agree with Dr. K. Singh said. It was too much. When we don't do anything, everything will improve. Livelihood of the farmer, what about national food security? Otherwise, greenhouses gas will go down, etc., etc. I'm really, that's my question. How weed infest that? Did you apply weedicide every year?
Sir, may I request you to question again, sir? Because
you said that stress of weed. I don't understand that. How did you apply weedicide as much as normally or more than normal, traditionally?
Okay, sir. For in the other years, beyond the study years, the recommended rate of herbicides were applied. But sir, during the recording of weed dynamics in the field condition, sir, I have recorded the observation from 2 by 2 meters per area from the each plot, and in that area, sir, during the 2 years, no herbicide application was done to simply see the effects of conservation agriculture on weed dynamics, like the effect of residue retention and three crop rotation along with minimum soil movement, how it can change the weed dynamics over the years. So in that area, in the demarcated area, we have not applied any herbicide to changes the weed dynamics.
But there must be some plot where there was no this crop residue management. Did weed flora change there also, in those plots also? Because
sir,
when we retain the crop residue, it
yes, sir.
suppresses weed control.
Yes,
How
sir.
about other plots? I many times feel, that's my apprehension, I may be wrong, totally wrong,
Yes,
that
sir.
crop, this conservation agriculture is being again promoted by weedicide industry.
Yes, sir. Sir, in the initial years of adoption, we have to rely on herbicides because of the challenges of weeds. But in the means long years of adoption, the because in zero tillage practices or in conservation, the weed seeds mostly 60 to 90% weed seeds remain in the 0 to 5 centimeter topsoil. So after the exhaustion of weed seeds over the years and with the recommended herbicide practices, means weed seed reduction could be achieved.
Let me share with all colleagues, when we initially started experiment with pesticide, which may become very limiting factor, how to control. But ultimately, after 8-10 years, it is the weed control. In organic farming, it is the weed which have become more important than the pest, as per PU experiments. Anyway, that is one caution. Then there was a question about adoption also. Do you know that area under FIRBs, furrow irrigated raised bed, that is again one component of conservation agriculture. It increased up to 6 lakh hectare in Punjab, in Haryana and Punjab. Haryana did better than Punjab. It was in early this century. It again went down. So how to popularize this conservation agriculture?
Sir, by a demonstration of the
Did a lot of demonstration with the money from CG system. In fact, Dr. I have forgotten the name from HAU gentleman, Dr. Yadav or something like that. He did a lot of work and Haryana made very good progress. Area under FIRBs was higher in Haryana than Punjab. Again, we went back. Then, okay, crop residue. How did you manage you then sowing of the next crop?
Okay, sir.
With maize residue, sowing was done by hand?
No, sir, by turbo seeder, sir.
How is it possible? If you have, did you shred the maize stover?
No, sir. It was in the residue return practices. It was cut about 60 centimeter height from the
So
base.
you shredded or you cut it?
Yes,
Then
sir.
how to do that at farmer's field, I don't know. But let me share with you the results of Punjab with the paddy straw management. New pests have come on wheat. Pink stem borer.
Yes, sir.
It is a rice pest.
Yes, sir.
It never came on wheat. And then one more is some worm. I have forgotten that. It is not. It is fall worm or something like that. Punjab and Haryana is water. Should we go for third crop there? We started with Pusa Baisakhi and G65 from Punjab. When I was student of IRA, Dr. S.S. Mass was head agronomy. He came with Pusa Baisakhi, PU came with G65. Then came in Punjab sunflower. Of course, it went up to 90,000 hectare and again came down. Then came spring maize. Now we are feeling that these crops have become. Should we go on popularizing now? This moong is very important crop. Spring season crop is very important in Punjab. What do you think as an agronomist?
Yes, sir. The crop, if we want the sustainable crop intensification, then we should involve one legume crop in the cropping system, sir.
That is fine, but what first limitation is water.
Yes, sir.
There is a report by Central Groundwater Commission and this Punjab State Water Resource Department. Report is of 2017 that in next 25 years most of the Punjab tubes will go dry.
Yes. Yes, sir. So then we can include any short duration crops, sir, having less water requirement, less water requiring crops, sir.
Very easy to say. How about livelihood of farmers? When we say that thing, I think we should also say that all those who are serving, their pay should also be cut by 5%. Sorry for this very harsh comment. Yeah, farmer also, I'm vice chancellor. My brother is doing farming. He also, my children are doing very good, have a good education. He also wants his children should have good education, good clothes, good transport facility. Anyway, so think about this. This is one question which is traumatizing, I will say, Punjab agronomist and Punjab scientists and Punjab government. How about third crop, which we are saying, and it is very important, particularly maize. Maize come as another rice crop, 20, 22, 25 irrigations, spring maize. Anyway, thank you. But there are many questions. We have to go to conservation agriculture, but how we have to do something. Thank you.
Thank you very much, sir.
Thank you, sir, for your insightful comments. So we'll move towards the next presentation, which is by Ms. Sunita from Discipline of Entomology.
Respected Chairman and jury members, very good morning to one and all. Myself Sunita, representing the Division of Entomology. I welcome you all to my presentation. Food security is often defined in terms of food availability and food utilization. Food security is achieved when sufficient quantities of food are consistently available to all individuals. If you look at the graph, India's food grain output and horticulture production touched an all-time high in 2021. However, the population will become 1.7 billion in 2050 and the demand for food grains will be 333 million tons while the horticulture production will be 650 million tons. But there are numerous factors that challenge the food production. One of the major challenges during crop cultivation is insect pests. Out of 10,000 pest species, major pests were nearly 10%, destroying 1/5 of the world total crop production and causes an annual investment of $40 million US. The first line of defense against pests by most of the farmers is insecticidal sprays. However, the indiscriminate usage of the pesticides led to many detrimental effects. At the same time, the demands underlying with crop production researchers from farming community and consumers are as follows. Farmers need sustainable, eco-friendly and safe crop production technology, while consumers need residue-free organic fruits, vegetables and grains. Then what is the way out? Yes, there is a way, that is biological pest control. Biological control is an effective means of reducing pests by using living organisms. Among the microbial biopesticides, Bacillus thuringiensis is well-known bacterial biopesticide since decades, but the commercial application of BT is limited due to susceptibility to temperature, humidity and UV rays. To overcome these bottlenecks, a hidden ecological role played by entomopathogenic bacteria as endophytes was sought. As the endophytes symbiotically colonize inside the plants, thus gets protection against abiotic stresses and UV rays, furthermore play a defensive role against insect-present diseases and also produce other benefits to plants. Background, the research was formulated and the title of research was Colonization of Entomopathogenic Bacteria in Brinjal Plant and Interaction with the Brinjal Shoot and Food Borer Lysinodus arbonalis. The objectives are evaluation of insecticides activity of native Bacillus species against Lysinodus arbonalis, standardization of inoculation techniques or methods to establish colonization of potential Bacillus species in Brinjal plant, to study the efficacy of colonized Bacillus species in Brinjal plant against Lysinodus arbonalis. Moving to the first objective, here the host insect has been reared for 2 to 3 generations and the units of third generation were used for in vitro bioassays. The method of bioassays in the form of spore crystal complex, that is 10 microgram per gram diet, and another form that is cell suspension, that is 10 power of 8 CFU per ml. And coming to the results, bioassays in the form of spore crystal complex showed that the highest mortality was observed in VTK HD1 strain with 100% mortality, followed by VKK13 and VKK-BB2 with 96% mortality, followed by VKK-BB1 with 80% mortality. The LC50 studies with the potential BT strains revealed that among native BT strains, VKK-BB2 was the most toxic with the lowest LC50 value, that is 0.59 microgram per gram diet. These are the typical gut symptoms produced in the neonates due to BT infection, and it has been proved as per Koch postulates. Bioassays in CFU form revealed that the reference strain BTK HD1 could produce 55.56% mortality, while BT strains VKK-BB2, VKK-13 and Brinjal seed endophytes BSCB6 and BSCB9 showed 51.85% mortality. And moving to the second objective, here the Bhagyamati variety was used for the pot culture studies and pots were laid out in complete randomized design and maintained in controlled conditions to find out endophytic ability of native Bt strains and the reference strain in brinjal seedlings via seed treatment, soil application and foliar spray. A single concentration of 10 power of 8 CFU per ml has been inoculated. The Bt colonies were prepared to arrive required concentration, that is 10 power of 8 cells per ml, for further pot culture studies. The re-isolated colonies were collected from the leaves, collected on 3rd day and 10th day after treatment. And coming to the results, the re-isolated colonies were subjected to morphological examination followed by spore crystal confirmation by phase contrast microscopy. Later, Chi-genes have been confirmed in these colonies via PCR techniques for confirmation to prove that re-isolated Bt colonies were of inoculated Bt strains. And with this, I can say that all 3 Bt strains were capable of translocation from seed to leaves and root to aerial parts of brinjal seedlings. And with respect to the colonization efficiency of Bt strains, here the VKK-BB2 was the potential colonizer in brinjal seedling in all the methods, as the highest recovery of VKK-BB2 was obtained in 2 samplings followed by reference strain BTK-HD1. Among all the inoculation methods, foliar spray was the best method followed by seed treatment in terms of number of CFU per gram leaf tissue. To visualize the Bt colonies inside the leaf tissues of the brinjal seedling, the potential colonizer Bt strain VKK-BB2 was used for the electrocompetent cells, and the transfer vector pCambia 1302 and these electrocompetent cells were subjected to the electroporation. And later, the positive Bt cells were examined under confocal laser scanning microscope for the green fluorescence in the positively tagged VKK-BB2 cells. And after the pCambia vector and GFP genes were confirmed in the positive Bt colonies, to proceed for pot culture experiments by using 10 power of 8 cells per ml in seed treatment, soil application and foliar spray. And coming to the results, here the confocal microscopy images of brinjal shoot and leaves colonized by GFP tagged VKK-BB2 cells indicated that the green fluorescence was detected in leaf tissues and stem tissues on 15th day after inoculation and 30th day after inoculations. The fluorescence was more on 15th day after inoculation, and green fluorescent cells were detected near the stomaton leaves. In soil application, both single dose and 2 doses in a week showed the fluorescence in leaf tissues up to 10th day after inoculation, and the fluorescence was found to be very low compared to the seed treatment. In foliar spray, in single and 2 inoculations per week showed the higher green fluorescence up to 15th day after inoculation in leaf tissues. And in the combination, it is soil application plus foliar spray in a week also indicated the bright fluorescence in leaf tissues up to 15th day after inoculation, while in the stem the green fluorescence was detected up to 10th day after inoculations. And moving to the third objective, here in the leaf bioassays, the neonates were released on leaves collected from Bt inoculated plants and control plants with 3 replications and 30 neonates for treatment. Coming to the results, the effect of inoculation methods could be noticed via microscopic observations, as the dead larvae were noticed on leaves from 5th day to 7th day after release on leaves. The percent corrected mortality was in the range of 23.33 to 44.44%, and effect of Bt strains on larval herbivory via seed treatment and soil application was low compared to the foliar spray and combination treatment. The reference strain BTK-HD1 showed the highest mortality, that is 44.44% in the combination treatment, and followed by native Bt strain, that is VKK-BB2 with 42.96% mortality. And these are some of the images were captured during the experiment. These are the dead neonates were collected on leaves after inoculations. And coming to the biochemical characterization of Bacillus species, carbohydrate test and antibiotic resistance studies proved that Bacillus species were diverse in nature. And with respect to the PGP activities, Bacillus species including Bt strains showed positive response to phosphorus and potassium solubilization in qualitative tests. Further, Some of these isolates exhibited extracellular enzymatic activities in addition to biocontrollability. The highest enzymatic activities indicated that the cellulose enzymatic activity was more in 3 strains, whereas the chitogenous enzymatic activity was more in Bt strains compared to the endophytic strains. Screening of these isolates for antagonism activity indicated that 3 neem leaf endophytic strains displayed antagonism against Alternaria alternata, while 2 neem leaf endophytic bacteria showed antagonism against Fusarium solani. And with this, I can say that establishment of indigenous Bacillus strain as endophyte might open a novel strategy to control the insect pests in a sustainable way. Coming to the conclusions, Bacillus species are potential entomopathogens. Bt strains as an endophyte in plant may become an alternative to Bt transgenic crop. In order to attain viable control of brinjal food borer, optimization of Bt concentration is required. Bacillus species possess multiple abilities which may help in colonization of host plants. And the way ahead is different inoculation methods of Bt strains to be explored further. Studies on interaction of Bacillus species with the plant physiology are very much essential. I am happy to inform that part of the research work has been published in Egyptian Journal of Biological Pest Control with the NAS rating 8, and one paper has been published in Pest Management and Horticultural Ecosystems with the NAS rating above 5. Papers were accepted in journals with NAS rating above 5. And I have participated in 2 conferences, received Best Poster Presentation at national level, and received Young Scientist Award in International Conference, and participated in the different trainings, and I have participated in extracurricular activities. And finally, I would like to express my sincere thanks to respective director and team, IRA, and my chairperson and advisory members, professor and head faculty members, Division of Entomology, and I extend special thanks to Dr. Dinesh sir and Dr. Lata ma'am for their support during the research work. And I wanted to thank PG School IARI, Dr. YSR Horticultural University, Andhra Pradesh, and family members and all friends. And I thank organizers for giving me this opportunity to present my PhD work on this prestigious platform. Thank you.
I'd like to invite Dr. Bansal for the interaction with the student.
Okay, Sunita.
Good morning, sir.
The idea looks all right. I think, it looks okay, promising, but frankly speaking, we have not reached there. In fact, from a PhD thesis, I would have expected, I could see that you did 2 objectives related to Bt and, brinjal, the problem you talked about at fruit and shoot borer. But then you later your third objective to, chitinase and antifungal activities, which was a little surprising to me. What was that? Why that required? Can you please tell me first? Your focus was, as per your title, only on fruit and shoot borer, right? Control, yes, which is important, no doubt about it, since we don't have Bt brinjal, transgenic, brinjal still approved in India. I would certainly look for the alternative mechanism you're talking about it. And this, as a biopesticide, has been known for ages, and that's how Bt came, the cryogens were isolated. But here the idea, why did you, in your third objective, from fruit shoot borer to fungal resistance?
Sir, actually, that is third objective is regarding that leaf biases against the neonids of the shoot and foot borer. And after that,
okay, fine. Sorry. No, that's fine. I can. I'll point as well. But why did you, go into measuring these enzyme activities and finding antifungal role with Bt? What was that?
Actually, sir, Bacillus isolates showing multiple abilities, sir. Actually, this Bacillus isolates for effective colonization in the host plants, they need some hydrolytic enzymes, sir. Here, I have done the cryogen confirmation for whether they have entered into the plant system or not. And after that, I have gone for the GFP technique. But if they have, suppose if they have the hydrolytic enzymatic activity like cellulase and xylanase or protease, then they get the chance to enter into the plant tissues. Then they get more effectively, they can colonize in the plants so that the problem will increase in the plants. Like with that idea.
Okay. I can understand. I thought that could have been done first. Actually, based on that, then you would have selected different strains and you would have taken them further. And what of these different strains, and why did you take only so many as you did? I don't know how many, 15 or so, whatever the number I couldn't capture. And where they were obtained from?
Sir, I have taken 25 bacillus isolates, sir. Actually, these isolates already earlier workers, they have isolated from the neem leaf as an endophytes, and even from the insects also, insect cadavers, the BT isolates, and even other than BT also, they have isolated the bacillus different species, like subtilis, amyloliquefacients, other isolates also are there. Then I have taken from the my labs.
And which cry gene did you use for confirming, the colonization of this particular bacteria and leaves? As you showed some PCR pictures. Which cry gene did you take?
Cry1, sir. Cry1, sir.
Which cry1A? Again, there are categories.
Yes, sir. Cry1A, sir. That is.
But then why you didn't identify in your slide?
Yes, sir. 276 base pairs, sir. That is
no, but why didn't you say so in your slide anyway? And then also coming to the question of mortality you finally studied, and yes. Again, two here. One is, why did you do it as a vegetative stage? I mean, I'm not an entomologist. I don't know when it gets, colonized. Will that be effective if it is at vegetative stage? You studied at vegetative stage, and what we are targeting is a fruit and shoot borer. You didn't yes. reach there at all. Number one, why the vegetative stage? And what was the level of mortality you mentioned was only up to 40 to 50% at the most. Yes, so sir, which is again, too low.
Yes, sir. The percentage mortality in the form of CFU cell colony forming units per ml, that is 10 to the power of 8 CLs per ml I have taken, sir. If you increase that concentration, means 10 power of 10 cells like that, the mortality may increase, sir. So actually, I have tried for the second objective also 10 power of 8 cells per ml for these inoculation methods. So the same concentration I have used for the bioassays also, sir.
No, I mean, your objective was to, I'm sure, to control fruit and shoot borer, right? Through the procedure you're talking about it, through this colonization yes, by the leaves. And so why didn't you, what were the problem? Why couldn't you reach to that level? Or was there any constraint of time or anything else?
Actually, I have done the experiments during that COVID time, sir, that lockdown time. And I didn't get time to extend that particular work after that June 2020, sir. So with that, I could do that work up to that level. So I have done that work. And here, being an employee in Dr. Vaisar Horticulture University, here my Honorable VC sir has given me a chance to extend my work here in the form of other projects, sir. So here
you're working elsewhere, not in IRI.
Sir, I am from Dr. Vaisar Horticulture University, Andhra Pradesh, sir.
Your voice is breaking.
Sir, I am from Dr. Vaisar Horticulture University, Andhra Pradesh, sir.
Okay, anyway, we'll have a discussion later, but thank you.
Thank you, sir.
Thank you, sir. May we have the comments from Dr. Patra, please?
Yeah. Thank you, Dr. Sunita.
Thank you, sir.
This is very interesting for me.
Thank you, sir.
Though I'm not, working on this area, not even, but BT cotton, we are very much aware, and also some work we have done about their impact in soil and other things. So my queries may be simple, very, very simple. What is the concentration of BT in soil? Have you ever studied the concentration of BT in soil? This is just a query.
No, sir.
And another thing is that you BT endophytes of brinjal, isn't it? BT in
yes,
the inside of the brinjal crop, your main goal, so that that will control the problems, the different insects of the brinjal crop. So in this, in the endophytes, when you are bringing something new, there must be native BT also in the brinjal. Have you seen their populations? You must have seen their populations. And what is that? How much it is? And what will be its impact on the other, endophytes of the brinjal? Because when you are bringing something, so there will be competitions. It may be, it may be synergistic. It may be antagonistic kind of things. So whether anything foreign things you are putting, so plant having its own mechanisms to throw it out. So it is my understanding, lateral understanding, So I like to hear from you, because ultimately that will be helpful. That will make the success. When something you are putting from outside and disturbing the natural ecosystems of the vegetations of brinjal, so there may be repercussions. There may be something which it will not easily adopt. So what kind of studies and information you have generated on that?
Sir, in the second objective, that is the main concept, sir. Actually, here we have done the 3 different type of inoculations, that is seed treatment, soil application, and foliar spray. The same concentration, that is 10⁸ CS per ml, I have given through seed treatment, and I have applied to the roots of these brinjal seedlings, and 15 days old seedlings and even 30 days old seedlings. Then I have gone for the foliar spray also, sir. And here I have observed that in seed treatment, on 15th day after inoculation, in the leaf tissues, the number of colonies were more compared to the 30th day after inoculation, sir. That means initially the BT has the ability to survive within the leaf tissues. In the seed along with the seed microbes also, it has given some competition and it has survived and it has gone to up to the leaf. But later on, that after that 30th day, the 30 days old seedling, that time the population were very low. So I have done the quantification of these BT colonies also in seed treatment and soil application. And to my surprise, sir, in soil application, when I have gone for the inoculation to the seedling, that is 15 days old seedling, it has shown higher number of colonies when compared to the 30 days old seedling. That means the plant has developed its own microbial community in the plant. So that is not allowing these foreign bacteria. But in early stages, that means the seed treatment and 15 days old seedlings, it is better compared to the 30 days old seedlings. Sir. And when compared to this foliar spray, it has more chance to enter into the leaf. Here, our target is that the neonates of this brinjal shoot and fruit borer, they have the unique behavior of the feeding cell. They will go inside. That is the borer, no sir. It will go inside. Whenever it hatches from the eggs, it goes inside the leaf and it eats on the internal tissue of that leaf, especially in the veins. So in that case, this endophytic works very well, sir. But here, the further it has to be explored, sir, and other type of inoculations. Like here in my study, I didn't do this root dip technique, sir. If we go for that before the transplantation of this brinjal, if we go for that seedling root dip in this BT solutions, it may give more effect, I think, sir.
Okay, thank you. All the best.
Thank you, sir.
Thank you, sir.
Can I have one more small clarification, please?
Please go ahead, sir.
Yeah, Miss Sunita, do you know about BT brinjal, the transgenic brinjal?
Yes, sir.
Where was the gene isolated from? Which strain of BT? Did you include that strain also in studies, by the way?
No, sir, I have not included BT.
No.
That is HT1 strain.
I will not ask you why. You should ponder over it, number one. Number two, again, since I know we don't have approval of this technology, but even then, scientifically speaking, do you think GM or transgenic BT brinjal will be a better choice, or your technology will certainly be useful and one can forget about BT technology?
My research definitely will give hope, sir. Definitely that if you apply the BT through seed treatment, because if you actually, sir, if you see the biofertilizers, here we are giving the biofertilizers as the seed treatment and application in the FIM in order to multiply, in order to increase the microbe in the soil. Like that, if you go for the
anyway, you're right, but we don't have results to say what you're saying.
Yes, sir. So if you go for the seed treatment with this BT, that is very easy, and we can.
No, but Madam Sunita, you have not shown any effect on fruit and shoot borer at the level,
sir,
from your experiment. So anyway, thank you.
Thank you, sir. So if there are no more queries, so then
Patel sir? No?
No, sir.
Anyway, so thank you, Dr. Dhuja. First of all, congratulations.
Thank you, sir.
And thanks to Dr. Sunita as well as your advisor. There's a lot of persistence to work with bioagents, because the effect of bioagent is slow compared to pesticides, or even when we talk of GM crops. A lot of extension effort is also required. It is just like allopathy versus Ayurvedic. We lost track of Ayurvedic. Now with COVID, we are again going back to Ayurveda. And about mortality, Dr. Bansal, now what in Punjab, most of, not only educated in villages, when they purchase brinjals from the market, they prefer a fruit where there is one hole of insect. That not too much of pesticide has been used. But I will come to your word indiscriminate use later on. How about when there is endophyte? Does it affect human health or not?
BT as an endophyte, sir?
Yeah, endophyte having a cry1 gene or that toxin.
So no, sir. It is safe to non-target organisms and even pollinators also. And even
I'm talking human health.
Human health also, sir. It is safe.
It is safe. Then
Yes, sir.
there should be no problem with the BT brinjal also. If it is safe.
Yeah, but in case of
someone should conduct experiment on it, maybe. Otherwise.
Yes, sir.
I have some other question, but I will not go in detail because there's something more important to share.
Yes, sir.
With all colleagues, with the faculty, students and faculty. We use what indiscriminate use. Undoubtedly, we are overusing and misusing. This is another word. We criticize ourselves. If we take large area, I think Punjab is the state. The coastal regions also, but if take the state on the whole, Punjab may be using maximum pesticides. If Dr. Mukesh is there, please correct me if there is any other state using more than Punjab pesticide. And situation is here.
Yes, sir. You are exactly right. The pesticides are being used more in Punjab.
Here is here BT has come. See how it has gone down and it is going down. Then everybody is saying that we are eating poison. Here, there are three things. I mean, three bars: blue, green, and red. Red is this pesticide residue above MRL, and blue is pesticide residue is there, but it is low, and red is above the limit, and green is free. See here, it was very bad situation in the 80s. It was more bad when we see the red above MRL, 44% of the food. And red plus blue is 92 and 8% is only free. And see the latest decade. It is decade-wise data. I don't believe in 1 year data. As I told in agronomy experiments also. And the doctor, I have forgotten the name, Dr. Ghosh probably, who worked on one location, but it was 2 years, so I kept quiet. It is altogether reverse situation. 92% is free. Only 1.37% of the food grain samples have a pesticide residue above residue limit. And the data are based initially on 1500, and here I think 6000, 7800. And those who are in entomology, there's no PAU slab on pesticide residue, one of the best in the world. So let us, you see, we submitted this paper. Dr. Mahapatra is also an author in that. Current science kept it for 2 years. I don't mince words. There is a lobby to criticize us, and we also use same words. And as we, now I am taking time. This is the, on organic carbon. Organic carbon is increasing in Punjab soils. See the green one. NPK consumption, it is stable during this decade. Now we published in PAU journal. Whoever is interested, I can send a copy of to that. 2 years, there is a lobby to criticize us, and we also use those very terms. Are not antibiotics being misused? No, discriminate use. During my stay in Germany for 9 years, only once I asked for antibiotic, and the doctor said, "No, paracetamol, you will die." I said, no, I will not die. We are accustomed to antibiotics. So let us have balanced presentation, balanced use of words. We have to defend ourselves. We use ourselves these words, then bureaucrats use those very words against us. My submission, please. Thank you.
Thank you, sir. So we can move on to the next presentation, which is by Ms. Rehene T from Floriculture and Landscape Architecture.
I welcome you all to my presentation. The dynamic shift of the country from sustenance production to commercialization, coupled with urbanization, has increased the use and demand for flowers. Flowers are quintessential in every aspect of social life. Among the flowers, rose is the #1 cut flower both in international and domestic trade. Roses have a plethora of uses, and they are pot plant, cut flowers, garden plant, loose flower, jam, perfume, essential oil, rose water, rose tea, and vitamin C source. The major rose growing states of India are Punjab, Haryana, Uttar Pradesh, West Bengal, Madhya Pradesh, Maharashtra, Karnataka, and Tamil Nadu, comprising of 29,000 hectares area and 30,000 metric tons production with export value of 47 crores rupees. Flawless superficial flowers are the criteria for its marketability, which are affected by diseases such as rust, powdery mildew, black spot, dieback, downy mildew, etc., among which phytoplasma diseases causing flower malformation and phyllo D are the most emerging diseases of the 21st century. With this background, my thesis was entitled Characterization and Management of Phytoplasma in Rose Genotypes. The objectives of my thesis are characterization of phytoplasmas using multilocus gene-specific primers in different genotypes of rose, identification of insect vector and weed alternative host responsible for natural spread of identified rose phytoplasma strains, and the third objective, management of rose phytoplasmas using antibiotics under in vitro and in vivo conditions. Surveys were conducted in 4 states of India, including New Delhi, Uttar Pradesh, Manipur, and Maharashtra. The disease incidence was recorded using this formula. Phytoplasma symptoms observed in rose cultivars are flat stem, phyllo D, flower malformation, leaf yellowing, and little leaf. Weed species collected from in and around the rose field were also observed to show phytoplasma symptoms such as witch's broom, stunting, little leaf, leaf yellowing, etc. Leaf hoppers that are putative vectors of phytoplasma transmission were collected from rose fields including Amraska, Empoasca, Exitianus, Ischimonus, and Orochius. The symptomatic rose, weeds, and leaf hoppers were collected and subjected to DNA isolation followed by PCR assays, agarose gel electrophoresis, sequencing, and analysis. Out of the 4 survey locations, the highest disease incidence was observed at New Delhi in the cultivars Hasina, Deepak, and MS Randhawa with a disease incidence of 87.5%. PCR amplifications of 1.25 kb was observed in rose, in weeds, and leaf hoppers by using the 16S rRNA gene primers. By utilizing the multilocus sec-A gene primers, PCR amplifications of 600 base pair was obtained in all the 16 rose genotypes. PCR assays with RP gene 16 SR2 group-specific primers led to the amplification of 1.3 kb product in 14 rows genotypes and 1.2 kb product in 7 rows genotypes with 16 SR1 group-specific primers. 5 rows genotypes showed amplification with both the primers, indicating a mixed infection. A multiplex PCR assay was developed, amplifying 2 genes in a single reaction utilizing 4 sets of primer pairs. Phylogenetic tree was constructed using 16S rRNA gene sequences and showed clustering of 12 rose genotypes, 6 wheat species, and 3 leafhopper species with 16 SR2 group. Phylogenetic analysis using SEC-A gene characterized 9 rose genotypes into 16 SR2D group and 7 rose genotypes into 16 SR1B group. Phylogenetic analysis of phytoplasma isolates using RP gene sequences showed clustering of 14 rose genotypes with 16 SR2D group and 7 rose genotypes with 16 SR1B group. The multiplex PCR assay was validated by phylogenetic analysis of the sequences obtained along with sequences obtained in normal PCR which clustered with each other in 16S rRNA gene as well as SEC-A gene sequences. The 16S rRNA gene sequences of rose genotypes obtained in the study showed similar RFLP pattern with 16SR2 group and 16SR1B group, except in the genotype Baramati1 showing different pattern at one enzyme indicating that it is a variant of 16SR1B. Virtual RFLP analysis of 16S rRNA gene sequences showed similar pattern with 16SR2D and 16SR1B in wheats and 16SR2D in leafhoppers. For in vitro management, the symptomatic cultivars were confirmed to be infected by phytoplasmas through PCR assay, and the cultivar MS Randhawa was selected after confirmation 60, 80, and 100 mg per liter of oxytetracycline, streptomycin, and erythromycin were filter sterilized and added to MS medium, and the explants were inoculated. The oxytetracycline treated plants showed significant differences in shoot length at initial stage. However, they regained normal growth and were free from phytoplasma as manifested by PCR assays. Whereas streptomycin and erythromycin treated plants failed to control phytoplasma in rows. For in vivo management of phytoplasma, the plant was regenerated by tissue culture and was transferred to the field, where antibiotic treatment was given, followed by PCR indexing. The oxytetracycline treatments eliminated phytoplasma from rows without any adverse effect. Salient findings. A survey was carried out in 4 states of India, of which the highest disease incidence of 87.5% was recorded in New Delhi. Mixed infection of 2 subgroups of phytoplasma in 5 rose cultivars was detected using the RP gene 16SR1 and 16SR2 group-specific multilocus gene primers. A multiplex PCR assay was standardized for detection of phytoplasmas employing 16SR RNA and SEC-A gene-specific primers in a single PCR reaction. 9 wheat species were identified as carrier of Candidatus phytoplasma asteris and Candidatus phytoplasma australasia, of which Coccinia species, Galensoga species are the first report in the world, and Poa annua is the first record in India. 2 leafhopper species, Hishimonas and Empoasca, were identified as putative vectors for rose phytoplasma strains. Oxytetracycline treatments eliminated phytoplasma in rose, and 60 mg per liter is optimal for elimination. Path ahead, surveys may be conducted in other parts of the country. Screening of phytoplasma-resistant genotypes of rose for its use in variety development. Environmentally safe management options may be explored, and standardization of meristematic culture for production of phytoplasma-free plants. I have one paper published in the journal Tree Biotech having a NOS rating of 8.4, the second paper in Tropical Plant Pathology having a NOS rating of 7.4, and the third paper in Propagation of Ornamental Plants having a NOS rating of 6.28, which is under review. These are the seminars I have attended. I was conferred Best Poster Award by Indian Association of Mycoplasmologists. I am also a lifetime member of Indian Association of Mycoplasmologists. Other activities include Best Article Award for Popular Article. I have also attended training for Japanese flower arrangement and published popular articles. I would like to acknowledge my chairman, advisory committee members, director, dean and joint director, IRI, Division of Floriculture and Landscaping, Division of Plant Pathology, National Fellowship for ST Students by Ministry of Tribal Affairs, family, friends and seniors. There are no miracles in agricultural production. Norman Ernest Borlaug. I would like to end with this quote. Thank you.
Thank you. So we'll like to invite Dr. Bansal for the interaction with the student.
Okay, thank you, Ms. Raine. I think good piece of work. But I feel you could have done more. As a PhD thesis, you could have done more work, actually speaking. Why the direction looks to be all right, of the work. But then again, you talked about in vitro management, use of antibiotics, right? So on what basis did you select these 3 different antibiotics? And which one you think was the...
Okay, so there were earlier reports that has been used in other crops that these 3 antibiotics are efficient in controlling phytoplasma. This is the reason why these 3 antibiotics are selected. And among the 3, oxytetracycline is the most commonly used antibiotic, and this was also in rose.
So are there reports of use of these antibiotics in rose, particularly for phytoplasma control?
No, sir. There were no earlier reports. It was in other crops, and that is why.
So which crop did you use it? In which crops did you then select these antibiotics from the literature? In which crops?
In other flower crops like chrysanthemum, fruit trees.
Against phytoplasma, right? And the phytoplasma strains which they were affecting, another, flowering plants, were they different in the strains than you identified, or any kind of similarity did you study? Can you hear me? I couldn't see you. Where have you gone? You're muted, please, Ms. Raine. Can you unmute yourself?
Yes, sir. Yes, sir. Yes, sir.
So did you find any similarity in the control of the phytoplasma in other flowering plants compared to the phytoplasma strains you isolated and you said they were newly, from in the world and one from India?
Yes, sir.
So what was
yes,
the
sir. In other crop as well, 60 mg per liter and 80 mg per liter was
No, I'm talking about
Madam
Raine, I'm talking about the phytoplasma strain. Was there any similarity? Are the same strains affecting other flowering crops and being controlled by the same antibiotics which you used in your rose plant as well? So without the particular knowledge you have generated in your thesis, couldn't you still control the rose phytoplasma based on the 3 antibiotics used in other flowering crops? How do you defend this?
Yes, sir. In other crops as well, it has been reported, but this is a study in rose that has not been taken up earlier, which is why to get an understanding, these 3 antibiotics was taken up. Besides, in India,
did you find? Okay, thank you. But did you find also any difference in the phytoplasma strains you identified in weeds or flowering plants of rose itself or in plant hopper? Were the same strains affecting these 3 different species?
Yes, sir. In my study, similar phytoplasma strains has been found in wheat as well as in the leafhopper, which is why we are concluding them as possible alternate host and putative vectors.
So what comprehensive management strategy would you suggest? Controlling plant hopper, controlling weeds, or controlling the infection in plant itself? The antibiotics you mentioned about it. So what's your message?
Yes, sir. The one control measure that will be very useful will be protected plant cultivation. And the second will be leafhopper control using insecticides or other environmentally safe options such as botanicals, including pyrethrum, rotinone, and other botanicals. And then also for weed control and removal of weeds and plant sanitation and hygiene in the field. And also the awareness among the farmers.
Okay, thank you.
Thank you, sir. Now we can have the question from Dr. Patra.
Thank you, Dr. Raine. Very interesting study and excellent information. It is a new area and we have tried. My query is, the management aspect of phytoplasma in rose. Rose is for aesthetics. And if we spray your, the antibiotics, oxytetracycline, and maybe number of spray you may need for that. I do not know whether that you optimized or not, but it may have the residual effect, and then if you put into the for the different occasions, so that the volatilization of that it may, I do not know, and the inhalation of that antibiotics may come into the our system and may cause the different kind of resistance and other things. So this is an issue about the management. Another is when we spray such antibiotics, maybe it will affect the other organisms, beneficial organisms. You have not worked, as I understand from your studies, but you must have come across its the negative impact on the different beneficial organisms in the ecosystems. So what are your knowledge on this? And of course, you have given in your future research that more natural way of management of these problems would be explored. But you have studied definitely in consultations with the literature and other things, but the negative aspects also you have looked into. So what is your opinion about this? Have you got my points?
Yes, sir.
Have you got my points?
Yes, sir.
So I like to.
Yes, sir.
So please give your response.
Okay, sir. Is better than cured. So I would suggest using clean propagation material. And if we're using from infected plants, then there is the option of culture. So these are the options, management options that can be taken up for prevention of this disease.
Okay. Thank you. All the best.
Thank you.
Thank you, sir.
Sir.
Any questions from the other expert members? I would like to welcome Dr. Ghosh, sir.
I'm glad to join this. Gain a lot of knowledge from the wonderful work done by most of the students. Thank you.
Thank you, sir. So if there are no more questions, I would request the chairperson, sir, for his comments.
Thank you, Dr. Dahuja. And first of all, welcome to Dr. Gaur. You may not be knowing, you are classmate.
Okay, sir.
I just 5 minutes earlier. Later, Dr. Sir, that you have joined.
Sir, wherever IR is there, I'm there.
Yeah. And thank you, Dr. Raine. Good work, good publications. And I want to compliment your teaching skills also. You're not fast like me and others. Student will appreciate you and your teaching methods, etc. You will become very good teacher if you continue in this field. Now, my question, I don't have many. Only one I will say that is just continuation of Dr. Patra and Dr. Bansal views or comments. And you could have gone further, at least with tissue culture techniques. You said standardization of that technique. Probably maybe you could not do that due to COVID, but that is the, I will say, easiest and cheapest way to control this thing, as Dr. Patra also mentioned about undesirable effect of weedicide, this antibiotics. I have one suggestion, if I have correctly understood, because my internet connection is not that good. MS Randhawa variety is one of the susceptible ones. So just when you said that Dr. MS Randhawa variety incidence of 87%, something like this, just came to my mind that we should not name varieties after the persons. Though it is a rose variety, but saying that the Emerson is most susceptible. I didn't, I mean, anyway, thank you. Thank you.
Bursa is even better. In nematology, people name nematodes after scientists, after nematologists, and then kill them,
Sir, as per the schedule, the lunch break time is 1:30. So can we take one more presentation?
It is up to the up to you and Dr. Rashmi.
Anil, we cannot continue.
No, we can take one. We can take. We will break. No, we can take, I think. Or okay, whatever.
Sir, it is not going to complete, no, sir. So let us have break.
We can
So we
smoke
come
up.
at 2:30, right?
Yes.
That's how
2:30
the field worker has to be very, very adaptive. Lunchtime 1 to 2:30, 3:30 to 4:30, 5 to 6, no matter. Leave it to organizer, Dr. Bansal. How does it matter?
No, it does matter because my food is not ready yet.
I see. then say that thing.
But you can have food for thought here.
So I would come back by 2:30 if you permit.
Yeah. It'll matter only 5 minutes, no problem.
Okay. As we did yesterday, yeah.
Okay.
So
Thank you.
So
See you.
can we break for the lunch now then?
Yeah. So
And we'll meet at 2:30. Okay.
2:30.
Okay.
Thank you.
Thank you.
Okay. Thank you, everyone.